There is a slight change in the program. Honorable Deputy Chief Minister, sir, is expected to arrive at 12:15 p.m. now, but we are starting with the proceedings in a short while from now. So I request you to please rise at your place when Deputy CM sir arrives. We'll start the programs in a few minutes from now. He is expected to arrive at around 12:15. Just अभी नहीं होगा अभी नहीं. I welcome all the dignitaries on the stage. I request Honorable Pro Vice Chancellor sir to accompany Dr. Pokhar Rawal, Dr. Lalit Mahatma, Dr. S.S. Chahal, Dr. D.R. Singh, Dr. C.D. Mai sir to accompany on the stage. Dr. Shalini Visayan ma'am, I also request you to please come on the stage. Please give them a big round of applause. We have Professor Dr. Pokhar Rawal, Secretary ISMPP, Rajasthan College of Agriculture on the stage. We also have Dr. S.S. Chahal, Emeritus President ISMPP, former Honorable Vice Chancellor MP UAT Udaipur on the stage. We also have as a guest of honor Dr. D.R. Singh, Honorable Vice Chancellor Bihar Agricultural University on the stage. We have as a guest of honor Dr. C.D. Mai, President, South Asia Biotechnology Center, New Delhi, Chairman, FC Limited, Mumbai, and Executive Board Member, ABNE, ISAA, USA, and President, Indian Society of Cotton Improvement, Mumbai on the stage. Please give them a big round of applause. It is with immense pride and deep sense of purpose that I welcome you all to the ISMPP 5th International Conference on Pathogens, Plant Health, and Food Security, Recent Advances for Climate Resilient Agriculture and Landscape Conservation, organized by Amity Food and Agriculture Foundation, Amity University, Uttar Pradesh, Lucknow campus, in collaboration with Indian Society of Mycology and Plant Pathology. It is with immense pride and great honor and deep sense of purpose, I extend a very warm welcome to each one of you to the conference. This prestigious conference is being organized by AUP and ISMPP. So before we start the conference, to set the tone for this remarkable intellectual congregation, I now have the privilege of inviting a distinguished academician and visionary leader, Professor Dr. Shalini Singh Visayan, Organizing Secretary and Director, AFAF, AUP, Lucknow campus. Ma'am, we request you to kindly present the curtain raiser and unveil the spirit and vision of the conference.
Good morning to one and all. As the organizing secretary of this prestigious conference and the director, MET Food and Agriculture Foundation, AUP Lucknow, it is a moment of immense pride and professional privilege to welcome such a distinguished gathering to the 5th International Conference of Indian Society of Mycology and Plant Pathology. We are deeply honored by the presence of our guest of honor, Dr. D.R. Singh, Honorable Vice Chancellor, Bihar Agriculture University, the legendary Dr. C.D. Mai, President, South Asia Biotechnology Center, and Dr. S.S. Chahal, Emeritus President, ISMPP, former Honorable Vice Chancellor, Maharana Pratap Agriculture University, Udaipur. We are delighted to have the leadership of ISMPP with us as Dr. Lalith Mahatma, President, Dr. Rawal, Secretary, Dr. Akhtar, Editor-in-Chief of the Journal of Mycology and Plant Pathology. On the behalf of our visionary, Honorable Founder President, Dr. Ashok K. Chauhan, Chairperson Dr. Asim Chauhan, Honorable Vice Chancellor Professor Dr. Balwinder Shukla, and Honorable Pro Vice Chancellor Professor Dr. Vashisht, I welcome our esteemed scientists, researcher from across the globe, and my dear student from every part of the India. The theme of today's conference, Pathogen, Plant Health, and Food Security, recent advances for the climate resilient agriculture and the landscape conservation, is not merely academic. It is a response to life reality. As the global temperature shift, the very behavior of the pathogen we study is evolving. New strains of fungi, bacteria, and viruses are migrating into the territories they never touched before. Our current agriculture practices are facing stresses that threaten the foundation of global food security. Today, agriculture must do more than adapt. It must become a tool for mitigation through smarter landscape conservation. Over the next 3 days, this conference is designed to be a collaborative engine focusing on the three pillars: leveraging genomic and digital tool for early pathogen detection, developing crop varieties and soil management techniques such as the bio-nanocomposite sustainable coating we see emerging today that withstand both biotic and abiotic stress. Integrating agriculture productivity with the preservation of natural landscape. There is saying that summarizes our mission today. That is very perfectly. The pathogen does not recognize the international border, and neither should our science. I encourage every delegate from the PhD student presenting their very first poster to emeritus professor to engage deeply. I firmly believe that the most of the significant breakthrough happens not just in the lecture theaters, but during a coffee break conversation or spontaneous debate during the technical session. So please fully utilize and enjoy your time here interacting with each other. I would like to express my deepest gratitude to ISMPP for honoring us to host this conference, our host institution, and the tireless member of the organizing committee who have worked behind the scene for the past 3 months to bring this vision to life. Most importantly, I thank my researcher. Your work in the laboratory and the field in the field is the front line of defense to this planet Earth. May your deliberation be fruitful, our connection be lasting, and the science be impactful. So, with this note, that pathogens know no border, science know no limit. Let us start with this conference. Once again, I welcome you all who are present offline, online, on the dais with me. I welcome you everyone. Namaste.
Thank you, Shalini ma'am. Organizing Secretary and Director, AFF, AUP Lucknow Campus. Now we'll start the proceeding by welcoming and felicitation of the dignitaries by Honorable Pro Vice Chancellor Dr. Anil Vashist sir and Dr. Shalini Viseen ma'am. First, I request to facilitate Dr. C.D. Mai, President, South Asia Biotechnology Center. Sir, please come forward. Please clap. स्वागत एवं सम्मान डॉक्टर C.D. माई सर का एक उत्तरीय पहनाकर और सालवर प्रेजेंट कर माननीय डॉक्टर C.D. माई का सम्मान करते हुए अब मैं रिक्वेस्ट करूंगा डॉक्टर D.R. सिंह, ऑनरेबल वाइस चांसलर बिहार एग्रीकल्चर यूनिवर्सिटी का विशिष्ट सम्मान करने के लिए, जो आज हमारे यहाँ विशिष्ट अतिथि के रूप में उपस्थित हैं। प्रोफेसर डॉक्टर अनिल वशिष्ठ और शालिनी मैम उत्तरीय पहनाकर आपका अभिनंदन करते हुए एवं विशेष मेमेंटो भेंट करते हुए स्मृति चिन्ह के रूप में आप करतल ध्वनि से स्वागत करिए। आपका इस सम्मान में आपकी ध्वनि, करतल ध्वनि भी सम्मिलित होनी चाहिए। अब मैं पुनः निवेदन करूंगा सम्मानित करने के लिए हमारे विशिष्ट अतिथि डॉक्टर D.R. सिंह का, ऑनरेबल वाइस चांसलर बिहार एग्रीकल्चर यूनिवर्सिटी, डॉक्टर D.R. सिंह, सॉरी, डॉक्टर S.S. चहल, Emeritus President ISMPP and former Honorable Vice Chancellor MP UAT Udaipur. माननीय प्रोवीसी सर और डॉक्टर शालिनी मैम उत्तरी और मोमेंटो देकर डॉक्टर S.S. चहल, Emeritus President का स्वागत करते हुए कृपया आप भी करतल ध्वनि से इनका स्वागत करें। आप वहां से करतल ध्वनि द्वारा ही स्वागत कर सकते हैं। Now I'd request Dr. Lalit Mahatma to felicitate Dr. Lalit Mahatma, President ISMPP. Honorable Provisi sir and Shalini ma'am to felicitate Dr. Lalit Mahatma. I'd request Honorable Provisi sir and Shalini ma'am to felicitate Professor Dr. Pokhar Rawal, Secretary ISMPP. हम विशेष रूप से आभारी हैं ISMPP के इस आयोजन में सहभागिता के लिए। उत्तरीय एवं मेमेंटो द्वारा स्वागत करते हुए प्रोफेसर अनिल वशिष्ठ सर और शालिनी विसेन मैम। Please give them a big round of applause. Now I'd request Professor Vasisht sir and Shalini ma'am to felicitate Dr. Jameel Akhtar, Principal Scientist, Division of Plant Quarantine, ICR, National Bureau of Plant Genetic Resource, New Delhi. Thank you very much, sir, for doing the honors, and give round of applause for this distinguished galaxy of scientists present on this stage. It is now my honor to invite a leader whose guidance continues to shape academic excellence and institutional growth. Professor Dr. Anil Vashist, sir, Honorable Pro Vice Chancellor, MIT University of Pradesh. Sir, we request you to kindly deliver the welcome address and formally welcome this distinguished gathering. Thank you, sir.
Greetings to Honorable Founder President, Sir, Dr. Ashok K. Chauhan, Honorable Chairman, Sir, Dr. Asim Chauhan, esteemed guest, Dr. C.D. Maiji, Dr. D.R. Singh Ji, Dr. S.S. Chahal Ji, Dr. Lalit Mahatma Ji, Dr. Pokhar Rawal Ji, Honorable Vice Chancellor, Amity University, Uttar Pradesh, Deputy Pro VC, Director, Amity Food and Agriculture Foundation, Professor Dr. Shalini Singh, Dean Academics, Director QAE, Head of Institution, esteemed faculty members, and dear students. I am honored to welcome you all to the inaugural session of the International Conference on Pathogens, Plant Health and Food Security, Recent Advances for Climate-Resilient Agriculture and Landscape Conversation, jointly organized by Amity Food and Agriculture Foundation, Amity University Lucknow Campus, and Indian Society of Mycology and Plant Pathology. We express our heartfelt gratitude to our Honorable Founder President, Sir, Dr. Ashok K. Chauhan, for his visionary leadership and dedication that has been instrumental in making Amity Education Group a beacon of excellence. Our gratitude to our Chairman, Dr. Asim Chauhan, for his utmost guidance and support. Sir's unwavering commitment to excellence has inspired a culture of creativity and collaboration on campus. Our gratitude to all esteemed speakers for being here with us today and sparing their valuable time. We are truly humbled and honored, and we say it with a huge round of applause. This conference brings together brilliant minds from across the globe, united by a shared commitment to addressing some of the most pressing challenges of the time. In an era marked by climate change, emerging plant pathogens, and increasing pressure on global food system, the importance of resilient agriculture and sustainable landscape conversation cannot be overstated. This conference provides a vital platform for knowledge creation, collaboration, and innovation in these critical areas. We are deeply privileged to have with us learned scientists, senior professors from across the world, who will be sharing their valuable insights, groundbreaking research, and diverse perspectives over the next 3 days. Their contributions will undoubtedly inspire meaningful discussions and pave the way for impactful solutions. To our researchers, faculty members, and students, this is a unique opportunity to engage, learn, and contribute. I encourage each one of you to actively participate, exchange ideas, and make the most of this enriching experience. Let us work together to foster innovation, strengthen global partnerships, and advance our collective efforts toward ensuring plant health, food security, and climate-resilient agriculture for future generations. My congratulations to Dr. Shalini Singh, Director, Amity Food and Agriculture Foundation, to Dr. Lalit Mahatma Ji, Dr. Pokhar Rawal Ji, and their entire team for their tireless efforts and dedication for successfully organizing this conference. I once again like to express my heartfelt gratitude to our Honorable Founder President, Sir, to Honorable Chairman, Sir, and all the esteemed dignitaries for sparing their valuable time to grace this occasion. Your gracious presence and constant support are a great source of inspiration to all of us and add immense value to this conference. Thanking you all once again. Jai Hind.
Thank you very much, sir. Now, I'd like to invite Professor Dr. Pokhar Rawal, sir, Secretary, ISMPP, to talk about the society and do felicitation of the dignitaries by ISMPP. Thank you, sir.
Thank you, sir. Good morning to all. Welcome, Abhinandan, Swagat, Vandan आपका ISMPP 5th International Conference में. Honorable Ashok K. Chauhan, Founder, President, Amity University, and Dr. Asim Chauhan, Chairman, Amity University, Lucknow Campus, Professor Balwinder Shukla, Honorable Vice Chancellor, Amity University, Professor Anil Vasisht, Honorable Pro Vice Chancellor, Amity University, Lucknow, and our today chief guest, Honorable Deputy Chief Minister Sri Kevasav Prasad Maurya ji, Uttar Pradesh. Today function, guest of honor, Professor C.D. Mai, our honorary fellow and past president of the ISMPP and president South Asia Biotechnological Center, New Delhi, and Chairman AFC Limited, Mumbai. Professor Asad Chahal, Honorary Fellow and Emeritus President of ICMPP and former Vice Chancellor, Maharana Pratap University of Agriculture and Technology, Udaipur. Dr. D.R. Singh, Honorable Vice Chancellor, Bihar Agriculture University, Sabol, Bhagarpur, Bihar. Professor A.N. Mukhopadhyay, sir, Honorary Fellow and past President of ICMPP. Dr. C. Manoracharya, sir, Honorary Fellow and Vice President of the ICMPP. President of the ICMPP, Dr. Lalit Mahatma, Professor and ADR, Navsari Agriculture University, Gujarat. Dr. R.S. Jaman, Professor, SDAU, Sardar Kushinagar, Gujarat, and Treasurer, Dr. S.K. Bhatnagar, Professor of MPUT Udaipur, Chief Editor, Dr. Jamil Akhtar, ICR, NBPGR, New Delhi, Senior Editor, Dr. Arup Kumar Mukherjee, Cuttack, Odisha, Managing Editor and Journal President of ICMPP, Dr. L.M. Suresh from Simiat, Nairobi, Dr. Sajina from Kerala, Dr. Sanjeev Kumar from Jabalpur, Dr. J.N. Srivastava from Bihar, Mohammad Yaqub from University of Kashmir, Pulwama, and our respected former President, Professor Naresh Mehta Sahab, our Chief Editor, Vice President of the ICMPP, and International and National Advisory Committee members, Organizing Secretary, especially Professor Shalini Singh Vijayan, Director MAT and Food and Agriculture Foundation and Head Funded Research Project here at MAT University, Lucknow campus, co-organizing secretaries, Professor Alpana Srivastava, Prachi Srivastava, Dr. Gurjit Kaur, and Dr. Komal Pandey, chairman and members of different technical sessions, publications, registration, accommodation, transportation, and food hall arrangement, finance poster exhibition committee members, international delegates, delegates from the different states of the country, distinguished guests, my dear students, person behind the curtains, ladies and gentlemen, and press and media personals. First of all, on my behalf and behalf of Indian Society and Mycology Plant Pathology, welcome you all in the inaugural session of 5th International Conference on a theme, Pathogens, Plant Health and Food Security, Recent Advances for Climate Resilience, Agriculture and Landscape Conservation. यानी रोगजनक पादप स्वास्थ्य एवं खाद्य सुरक्षा, जलवायु अनुकूल कृषि एवं भूदृश्य संरक्षण में हालिया प्रगति विषय पर आयोजित हो रही है. We all are grateful and I extend my warm greetings and thanks to Dr. Ashok K. Chauhan, Founder President, MAT University Group, and Dr. Asim Chauhan, Chairman, and Dr. Barvinder Shukla, Madam, Vice Chancellor, Professor Anil Vasis, Sir, Pro Vice Chancellor, and Professor Shalini, Madam, Director MAT, for providing an opportunity of a organization of this international conference of ICMPP here. Thank you very much, Madam. The Indian Society of Mycology and Plant Pathology was founded in the year 1970 by the eminent plant pathologists of the country, Dr. N. Prasada and R. Prasada, who felt necessity to publish the research finding without delay in journal during the formative stage of the society. Academician and industrialist Dr. P.P. Singhal, founder of Messrs. Pesticide Industries India, Udaipur, helped the genius way in various ways. He was founder patron of the society and instituted an annual award, Pesticide Industries Award. The first issue of the Indian Journal of Mycology and Plant Pathology was released in December 1971. Initially, the journal published twice in a year. Later on, from 1982, its frequency was raised to 3, and from 2010, 4 issues per year is publishing. Since then, the journal has been regularly published and in a wide circulation in India and abroad. The society has also started publication of ICMPP newsletter. Society endorsed for a new and unusual venture of taking responsibility of beginning a publication of annual review of plant pathology from the year 2002. The other activity of the society include the organizing annual conferences and journal meets. The society has sponsored in the all 42 annual conferences till date, covering almost all the states of the country, 4 global and international conferences at Udaipur and Jaipur, 3 Asian conferences at Mysore, Hyderabad, and SDAU Sardar Kushinagar were successfully organized by the society. In recent years, society is engaging organization of journal conferences by the respective journal presidents. The regular feature of the annual conferences are different activities such as YLN Outstanding Plant Pathology Teacher Award, SC Dubey Outstanding Young Scientist Award, Professor S.H. Charles Lifetime Achievement Award. The society also holds the award competition for PP Single Memorial PI Industries Award, Srimati Guman Devi Verma Memorial Best Women Scientist Award, PR Verma Award for the MSc and PhD Plant Pathology students, and Dr. K.S. Bilgrami Best Poster Presentation Award, and R. Prasada Best Published Research Paper in Journal of Mycology and Plant Pathology. In addition to these awards, it includes the Presidential Address, N. Prasad, R. Prasad, BP Bide, YL Nenne, PR Verma, and Bibel Thakur Memorial Award lecture, also by eminent plant pathologists of the country. The society has in all more than 1,200 members and journal subscribed by the all agriculture university and traditional university in India and subscribed by 20 different countries. Over the past 15 years, ISMPP has achieved multi-phased growth, particularly through the high quality scientific research via a flagship publication, the Journal of Mycology and Plant Pathology, nowadays upgraded on a online editorial work and indexed in the Web of Science and NAS. JMPP is available now online and students and scientists can download any contents. The society has thus come a long way and our sincere effort is to take it much ahead. We look forward to our patrons, peer office bearers, and all the members to continuous support and encouragement. A key objective of this conference is deliberate on how enabling policy framework and institutional support and empower rural communities and enhance agriculture sustainability. The conference will also highlight agroscaping as well as transformative tools for disease management and food security. Core theme including climate adaptive cropping system, ecosystem restoration, rural landscape transformation, flood resistance, agriculture design, and cultural dimension and agrarian landscape, emphasizing the importance of holistic and sustainable land stewardship. By the integrating digital innovations, smart technologies, and ecological design principles, the Congress aspired to bridge traditional wisdom with the frontier science. ISMPP, under the aegis of MIT University, provide a platform and forum for the interaction among the researcher, impressive deliberations, and sharing idea to arriving to some conclusion, apprehension to the crop disease and their sustainable management through new and innovative approaches in the plant disease research and development of trained human resources for food security. I consider it a great honor and extend heartiest welcome to all of you, all of delegates, and my best wishes for a grand success of the international conference. Jai Hind, Jai ISMPP. Besides that, now it is matter of pleasure, all of us, to felicitate on behalf of Indian Society of Mycology and Plant Pathology. So, first of all, now first I invite Dr. Professor Anil Vasis sir to have a felicitation from the side of yes, our I request our CDMI sir to present a honor to Dr. Anil Vasis. हमारे ये अभी CDMI का तो करो करवा दीजिए Now I request our Pro Vice Chancellor Anil Vasis sir to honor our respected CDMI sir. Professor, yes sir. कौन हाँ ये दोनों रहेंगे ऐसे चल सर Now I request our President ICMPP, Dr. Lalit Mahatma sir, to felicitate our respected Professor S.S. Chel sir, Professor Chel sir. अपने बिहार वाले सर का
बिहार सिंह का एंड
देन कौन बच गए प्रेसिडेंट आईसीपी ललित माधव Now I request our President Emeritus Professor S.S. Chel sir to felicitate Dr. D.R. Singh, Honorable Vice Chancellor, Bihar Agriculture University, Bihar. फिर महात्मा सर का जमीन सर पर हाँ फिर वो प्रेसिडेंट ललित महात्मा जी को करते हैं I also request our child sir felicitate Dr. Jamil Akhtar. Dr. Jamil Akhtar, please. ललित महात्मा से भी करना पड़ेगा हाँ
ये विशेष सम्मान आईएसएमपीपी द्वारा किया जा रहा है और जो ये राजस्थानी पगड़ी है ये विशिष्ट रूप से सर लेकर के आए हैं राजस्थान से। करतल ध्वनि से स्वागत करें। उत्तर प्रदेश और राजस्थान की संस्कृति और पूरे भारत की संस्कृति का समन्वय है।
Thank you, sir. Now I request Honorable CD sir, CD my sir, to felicitate our President of ICMPP. हो गया?
नमस्कार मित्रों, मैं परम आदरणीय सीडी माई साहब से निवेदन करता हूं कि हमारे यंग और डायनामिक सेक्रेटरी को सम्मान करें साफा पहना के। मैं सीडी माई साहब से इस कार्यक्रम के आयोजन के लिए जो सारा भार संभाला है, ऐसे ये इतिहास में पहली बार है कि कोई महिला ऑर्गेनाइजिंग सेक्रेटरी बनी है इस सोसाइटी के किसी प्रोग्राम की। महिला वाइस प्रेसिडेंट बनने जा रही है और इतना ही नहीं, अगर मान लो आप पूरा प्रोग्राम देखेंगे तो जितने भी ऑर्गेनाइजिंग और को ऑर्गेनाइजिंग सेक्रेटरी ये सारी महिला है। उन सभी के बिहाफ पे सम्मान लेने के लिए मैं वाइजन मैडम को भी आमंत्रित करता हूं। मैं वापस रिक्वेस्ट करता हूं सीधी भाई साहब से कि इन दोनों यंग और एनर्जेटिक लोगों का सम्मान करें। धन्यवाद।
करतल ध्वनि से स्वागत करें। सम्मान हमारी उस परंपरा का वाहक है जो हमें प्रेरणा देते हैं पुनः पुनः उसी कार्य को और उसी तरह के विशिष्ट कार्यों को बार बार करने की। आप इस सम्मान में विशिष्ट डॉक्टर शालिनी विसेन सम्मानित होते हुए आईएसएमपीपी द्वारा। ये सही बात है कि सर के सम्मान में आपका भी सम्मान निहित है, पर आप भी सम्मानित होंगे सर। प्रोफेसर रावल सम्मानित होते हुए, प्लीज गिव हिम अ बिग राउंड ऑफ अप्लॉज। और मैं पुनः प्रोफेसर रावल को यहां आमंत्रित करूंगा कि वो मंच पर आ जाएं।
मैं अब सीडी मई साहब से निवेदन करूंगा कि हमारे वर्तमान प्रेसिडेंट डॉक्टर ललित महात्मा सर का सोसाइटी की तरफ से स्वागत करें। अब इसी मंच पर हम आईएसएमपीपी की तरफ से फाउंडर प्रेसिडेंट ऑनरेबल डॉक्टर अशोक चौहान साहब एंड चेयरमैन डॉक्टर असीम चौहान साहब एंड वाइस चांसलर डॉक्टर बलवीर शुक्ला का भी स्वागत अभिनंदन करना चाह रहे हैं। तो अभी हम हमारी तरफ से एमईटी यूनिवर्सिटी को उनकी अनुपस्थिति में आपको हैंड ओवर कर देंगे। थैंक यू सर, थैंक यू वेरी मच।
Thank you very much, sir, for doing these honors. And once again, I request for this joint effort, please give them a big round of applause to ISMPP and to AUP Lucknow campus. Now I'm going to call an eloquent speaker. You have already met him. Our special invitation to Dr. Lalit Mahatma, sir, President ISMPP, to address this August gathering.
नमस्कार। गजल के वास्ते शब्दों का मिजान रखते हैं। धधकते दिल में स्वागत का लोबान रखते हैं। इन अमेठी वालों का क्या कहना, इन अमेठी वालों की मेहजबानी का क्या कहना। ये है अहले लखनऊ, एक अलग पहचान रखते हैं। धन्यवाद अमेठी। परम आदरणीय उप मुख्यमंत्री श्री केशव प्रसाद जी मौर्य, जो वर्तमान में तो नहीं है लेकिन हम सभी के दिलों में यहाँ पर हैं और बहुत जल्दी अपने बीच में उपस्थित होने वाले हैं। परम आदरणीय अमेठी यूनिवर्सिटी के फाउंडर प्रेसिडेंट अशोक चौहान साहब, अमेठी यूनिवर्सिटी के चेयरमैन परम आदरणीय असीम चौहान साहब, अमेठी यूनिवर्सिटी के ही वाइस चांसलर डॉक्टर बलविंदर मैडम, आईएसएमपीपी के सिपे सालार, जिनका कोई परिचय की जरूरत तो नहीं है लेकिन कुछ लोग जो इस डायस की शोभा बढ़ा रहे हैं, उनमें से परम सम्माननीय डॉक्टर सीडी माई साहब, परम सम्माननीय डॉक्टर एसएस चहल साहब, बिहार एग्रीकल्चर यूनिवर्सिटी के प्रसिद्ध ऑनरेबल वाइस चांसलर डीआर सिंह साहब, प्रो वाइस चांसलर वशिष्ठ साहब, अनिल वशिष्ठ साहब, शालिनी मैडम, हमारे एनर्जेटिक एडिटर इन चीफ जमील अख्तर साहब, मेरे मित्र सेक्रेटरी डॉक्टर पोखर रावल, और जिन लोगों का नाम उन्होंने अपनी स्पीच में लिया है उन सभी लोगों का नाम मैं लेना चाहता हूं दिल से, लेकिन मैं किसी का नाम दोहरा नहीं सकता। अगर पंक्ति में मैं देखता हूं तो बड़े माननीय लोग बैठे हुए हैं, और चूंकि इन सबको बीमार पता है कि मुझे डायस पे चिपकने की बीमारी है, तो मुझे कम से कम 5 बार इन्होंने कह दिया गया है कि आपको 2 मिनट में खत्म करना है। तो मैं 2 मिनट में खत्म करने के लिए आपको सिर्फ एक शब्द बोलना चाहता हूं कि दोस्तों, जब मेरा प्रेसिडेंशियल एड्रेस देने की बारी आएगी तब मैं आप लोगों को अंग्रेजी में विज्ञान की बातें समझाने की कोशिश करूंगा। लेकिन फिलहाल मैं सिर्फ इतना कहना चाहता हूं, मेरे जितने भी स्टूडेंट्स और यंग मित्र बैठे हुए उन सभी के लिए, कि सत्य पर अड़े रहे, सत्य पर अड़े रहे, किसान के साथ खड़े रहे और सोसाइटी के साथ जुड़े रहे। आप सभी का बहुत बहुत धन्यवाद।
धन्यवाद डॉक्टर पोखर रावल साहब। जैसा कि आपने कहा कि आप I told you earlier that he's an eloquent speaker, and a special mention for his praise which he did about Amity University. And I would like to mention that you are in Lucknow, and I would like to say one एक शेर मुझे याद आ रहा है, लखनऊ है तो महज गुंबदों मीनार नहीं, लखनऊ है तो महज गुंबदों मीनार नहीं, सिर्फ एक शहर नहीं, कूचा व बाजार नहीं, इसके दामन में मोहब्बत के फूल खिलते हैं, इसकी गलियों में फरिश्तों के पते मिलते हैं। और ये शहर जब धड़कता है दिल बनकर, इसकी आबो हवा सांसों में समा जाती है, तब उतर जाती है वो रूह की गहराई में, बन के तहजीब वो लोगों को नजर आती है। इसी लखनवी तहजीब से मैं पुनः आपका स्वागत करता हूं, और एमिटी में विशेष स्वागत, जहां तहजीब और आधुनिकता दोनों का समन्वय एमिटी विश्वविद्यालय। और इसकी क्रम में नाउ I'd like to invite our guest of honor, Dr. S.S. Chahal. It's a special privilege that we have Dr. Chahal, Emeritus President, ISMPP, and former Honorable Vice Chancellor, MP, UAT, Udaipur with us. I humbly request Dr. Chahal, sir, to address this august gathering. Thank you, sir.
Honorable Chancellor Dr. Ashok Chauhan ji, Honorable Chairman Dr. Aseem Chauhan ji, Dr. C.D. Mai, Dr. D.R. Singh, Dr. Anil Vishisht, Dr. Lalit Mahatma, Dr. Rawal, Dr. Shalini Singh, Dr. Jameel Akhtar, Professor Mukhopadhyay, Dr. Manoj Acharya, and dear participants, ladies and gentlemen. First of all, I'm very much thankful to the organizers who have given me this opportunity to share my words with you on this very important occasion. Personally, I'm closely associated with the activity of this society, ISMPP, for the last 45 years. And I'm happy that this international conference that is being organized as planned by the society to hold such conferences after every 5 years. The conference has special significance in the sense that this is an occasion to assess what we have done, what we need to do in future. And we get directions for our youngsters how to shape up their studies, their research, their teaching. This is high time to identify, of course, action and set priorities for plant pathologists. What seems to be important at this occasion, to my mind, is that besides This importance of risk of invasive species, exotic diseases, which we are facing, host-pathogen interaction, biological system, genomic, molecular, ecological approaches. It is desirable to investigate relations on climate change, which is we are facing all in all the spheres. And this has very nicely been taken up as a thematic area in this conference. Climate change, of course, is a major driver affecting all this triangle, fearing always the emergence of new virulences, and it attacks forces and multiplier of plant diseases and pathogens and their evolution, and it enhances their survival. We have made, of course, tremendous progress in various fields of agriculture. We all know that this is one area where country can take proud that just 50 million tons of food production at the time of independence in 1950. Now we have 357 million ton of food grain and 362 million ton of horticulture produce. This is a success story in itself. And in this success story, of course, the plant pathology has a special significance containing crop losses from pests and pathogens. But we need to remain ahead of pathogens for which such conferences are being organized in a technical way. It's a matter of course, notice that our large national agriculture research and extension education system, the scientists have been continuously generating data, recording massive data. And for this reason, because we have a tremendous data, this has helped the country scientists to take this country ahead. And in this way, we have taken a leapfrog in artificial intelligence bandwagon very quickly, and this which has not been taken up by many countries so far. So this is how different areas, these are being addressed in this conference. When we are talking of advanced technologies, in this, of course, machine learning, that is, of course, we are noticing it is coming up. Autonomous machinery in agriculture, that is coming up. All our youngsters, they are taking notice of it. And AI altogether, it is changing plant diseases and diagnostics and decision support system. So there is tremendous scope of this expansion of this area in future. And this is always, it's a landscape of agriculture, in fact, that is under transition phase. And there are new courses of AI. These are being developed. New centers are being planned in different universities in this specific area to develop AI tools. What we hear in this respect, I will share my thought in this, that what we hear, and this is misplaced consideration that AI, that will surpass the human minds. Because AI tools, so far, they have been made to answer, but they cannot question. So far as questioning is concerned, it remains the domain of human mind, and that will never be taken off from this. Innovation comes from human mind. Second is that we also fear that AI that will shadow the employment. But look at the computer technology, which at the time of introduction, it was largely propounded that it's employment eater. But what happened? It turned up otherwise. One of the major employment generator technology that is computer now. Similarly, in agriculture is, and even agriculture-oriented agriculture will remain as major employment provider in the sector of our agrarian system in the country. In light of such developments, there is need to emphasize research on detection of diagnostics, capacity of this pathogen identification, developing new cultural practices, new eco-friendly approaches. Participants who are attending this conference, they will take note of it. Genetic engineering, disease resistance, strengthening decision support system, formatting new location-specific IPM modules. I must share with you that this program has been very meticulously prepared. All the technical sessions are there. And it is expected that this conference will come up with valuable recommendation from this will emerge, which will be able to send to different policy planner and Indian Council of Agriculture Research to take note of this for the benefit of our researcher, teachers who are working in different institutions. With these words, I thank you all. I thank the participants who have come from various institutions, students who are here, who are associated with agriculture. And once again, I thank the organizers who have organized this conference here in Amity University. Thank you very much.
Thank you, Dr. S.S. Chahal, sir, Emeritus President, ISMPP. Now it's my privilege to invite Dr. D.R. Singh, sir, a guest of honor today, Honorable Vice Chancellor, Bihar Agricultural University, to the dais. Thank you, sir.
Well, the Professor C.D. Mahi, the former chairman of SRB, New Delhi, Dr. Asim Chauhan, Dr. Asim K. Chauhan ji, Dr. S.S. Chahal, the former Vice Chancellor, Udaipur, Dr. Lalit Mahatma, Dr. Pokhral Parwal, Dr. Anil Basist, and the very important, the International Conference, the organizing secretary, Dr. Sanli Singh. First of all, let me thank the organizer for giving me this opportunity to be associated with this important international conference. And my special thanks goes to the organizers from the Amity University, and more particularly to the Dr. Sanli Singh, organizing secretary. Well, friends, I am coming from the state Bihar. As you know, Bihar is the fastest growing state of the country as of now. And also, you may be aware the three things, important things from Bihar. The lychee is known from Bihar, and the makhana is also known from Bihar. So why these things are very popular? We from the Bihar Agricultural University, we obtained 5 GIs for our country. Mithila makhana is the one, Shahi lychee is the number 2, and Katarni rice, Maghai pan. These are the important GIs we obtained, and Jardalu mango, 5 GIs. And these 5 GIs of the product being exported from Bihar to the entire part of the country, also to other states. Why I have been telling this? Because how this important, the GI for the nation, as well as important for this galaxy of the pathologists gathered here. The Maghai pan, which we eat taking in the state of Uttar Pradesh, that is a GI obtained from Bihar, not from the Uttar Pradesh. So in a process, we have identified around 30 more products from Bihar to obtain the GI. And very soon, we are going to have another GI for the Litti Chokha, as well as also the Sita Sindur from Bihar. So this is very important. As of now, what being exported from the Bihar as agricultural products, the more importantly, the Jardalu mango, and the Shahi lychee, Katarni rice, and the Mithila makhana. So in all these 5 GIs, our university, the Bihar Agricultural University, has played a major role in obtaining GI, and thereby its population in the country and the outside the country. And as you know, the Bihar is the only state of the country where we get 85% of the production of the makhana from the Bihar region. So then for this food safety and food security, the importance of pathologists is very important. And to export any commodity, agriculture commodity from Bihar or any other states, the scientist has to play a major role for its proper package and practices, its safety standard, the proper packaging and the branding. Whereas in all these issues, the pathologists are very important. See, in this context, I believe this important international conference being organized at this university, it's a very right time. Historically, the essence of the discipline of the plant pathology has been very well emphasized on the three major occurrences. The first is the occurrence of the rust epidemics in wheat, as you all know. And second is the late blight of potato that has significantly affected our food systems and economies of the worldwide. And third is very important, in India, the occurrence of the Bengal famine during the 1942 and 43 due to the widespread of outbreaks of brown spot disease of rice. So these three disease outbreaks remind us how crop failures can trigger humanitarian crisis and also losses to the livestock. So this natural disaster resulted in the realization of the importance of the plant pathology and initiated a systemic study in this discipline across the country. So these losses are further compounded by the climate change. The modern agricultural landscape is increasingly shaped by existence of simultaneously biotic and abiotic stress. In the scenario of the climate changes, we faces a lot of challenges that is directly or indirectly that affects the plant physiology, weakening natural defense system, and may lead to breakdown of resistance in the crop. In addition to that, the pathogens are mitigating to new regions, expanding their host range, adapting to changing climatic conditions, and evolving into more aggressive forms. As a result, we are witnessing the emergence of new diseases and resurgence of pests. Notable examples include the fusarium head blight in wheat, which is spreading to new geographical locations and increasing the severity of diseases. The emergence of the tropical race 4 in banana is another challenge threatening the banana industry globally. The threats are equally applicable to Indian banana industry with a record production of banana ranking first in the world with the nearly 25% of the world banana production. Similar milestones have been set in many other crops like millets, rice, mango, papaya, lemon, and spices as per the recent recommendation of the Ministry of Agriculture, Government of India. Well, as we proceed towards food security, the focus of agriculture is gradually shifting from food sufficiency to nutrition security and food safety with quality produce that are nutrient rich in residue free. Many of the pathogens associated with the food produce, toxins like aflatoxins, etc., and the contamination of foods with mycotoxins poses serious health risk to both human and livestock. So in this context, there is a pressing need to adopt innovative methods and integrated solutions to address new challenges in plant health management. So I believe this conference is very timely when agricultural landscape is facing multiple complex challenges in a way to meet out these desired targets. It covers a wide aspect and relevant themes for optimization of crop health, particularly against plant pathogens. I believe the conference would provide a very constructive platform to all the stakeholders involving the students, the scientists, the policymakers, and the educationists for fostering the meaningful discussions and would pave way for a collaborative efforts towards protecting plant health and strengthening food and nutrition security. With this, I wish the organizing team for a grand success of the seminar, and my special thanks goes to the Amity University and more particularly to the Dr. Salini Singh for inviting me to be the part of this very important international conference. Thank you. Jai Hind.
Thank you very much, sir, for giving a new dimension to the seminar. And as sir mentioned about the Bihar's inclusion of different new things, including Litti Chokha, especially I would like all of you to clap for Litti Chokha and other inclusions, which will give a recognition which is due for a very long time. इसमें होता क्या है कि हल्दी हमारी है और उसका जियो टैग और बाकी चीज अमेरिका में हो जाता है. So it's highly desirable that we recognize that whatever the tradition and culture we have to bring it forward to the world. Now, it's my privilege to invite Dr. C.D. Meiser, President, South Asia Biotechnology Center, New Delhi, and Chairman, AFC Limited, Mumbai, Executive Board Member, ABNE, ISA, USA, and President, India Society and Cotton Improvement, Mumbai. C.D. Meiser is the guest of honor. It's my privilege to invite sir. Thank you, sir. Please.
सर्वप्रथम मैं अमिटी यूनिवर्सिटी से हमारी जो शालिनी जी है उनका बहुत बहुत आभारी हूँ. बहुत बहुत धन्यवाद करता हूँ कि उन्होंने इस कैंपस में मुझे बुलाया. अमिटी का मतलब मैत्री संस्कृत में होता है और वो जो मैत्री आपने दिखाई उसके लिए मैं बहुत बहुत आभारी हूँ. And when I entered the campus yesterday, a large number of students around me, I felt the odor of academics. I think it's very rare आजकल कैंपस में जाने के बाद nobody discusses science. They discusses more on politics. But when I came here, I felt that people are discussing academics. So I congratulate Amity for wonderful atmosphere created in this particular campus for students. आपको students को भी मैं बधाइयाँ देना चाहता हूँ because they are learning what is important. I think I would first Professor Vashishtha, then Lalit Mahatma ji, Pokhar ji, हमारे मित्र Dr. Chahal. We have on the dais Secretary and President of the Society. I have lots of friends in the audience, Dr. Mukhopadhyay, Dr. Manoharachari, and so many others. एक संस्कृत में कहावत है, पात्र विशेषे न्यस्तम गुणांतरिम व्रजति शिल्पमाधतु जलमेव समुद्र शुक्लो मुक्ता फलसी पयोदस्य. इसका मतलब यह है कि a small drop of water, if it falls in an ocean, it becomes salt. And in the same drop of water, if it falls in the mouth of a snake, it becomes poison. But the same drop of water, if it falls in an oyster, shimpla, I don't know what they call it in Hindi, shimpla, we call it as oyster, it becomes pearl, moti ban jata hai. I think the most important thing in science or our all efforts are that we must see that whatever perspiration which we do falls in the oyster so that it becomes pearl. हमारे जो community है, plant pathology community, they have been struggling for so many years to really management of how agriculture could be more productive. And how much they have contributed, I think my earlier speakers, Dr. Chahal, Dr. Singh Saab has already narrated you. That the simple epidemics of wheat rust was how was managed by only what is called as disease-resistant varieties. How, for example, the current problems of agriculture, like maybe it is a T4 race in banana, or maybe it is a pest in cotton, or maybe it is some other diseases which we are facing. They're all being managed. So it's a continuous process and a fight between what is happening between pathogen, the host, and then on the third side, we are as a scientist. And probably you will see that large number of papers which you have now gathered, they're all related to basically how management is done. I think this is very important, and this particular conference, I'm sure that we are all going to talk about pathogens. We are going to talk about the variability of pathogens, food security of the nation, and whatever little advances which we are facing. Let me tell you, agriculture in the current scenario is facing the biggest problem is of climate change. And I think you all very well know that because of uncertainty in the climate, we have new problems which are emerging. Climate change is one where we can mitigate somewhere, but there are some, what we call it as unprecedented problems that we are facing. For example, the current war between Israel, USA and Iran. Now, this was not expected. So this is what exactly now this war will have implications on Indian agriculture. I am not talking about pathology alone, but what implications they have in agriculture are very important. I think the most important part is, somebody was telling me yesterday that we are going to have shortage. 45% of our nitrogen fertilizer comes through the Strait of Hormuz. So naturally, it's going to be a problem for our farmers to get timely, probably input in the coming Kharif season, which is one of our most important seasons. Now, it is also predicted that all this, not only because of war, but El Nino is going to set late in this season. Probably you must have already read the reports of meteorological department where they have said that probably now there is a El Nino factor. El Nino means it is the ocean waters are not cooling. They are cooling now, but it takes time for them to really get hot. And unless the surface get waters hot in the ocean, monsoon will not start. And that is what we call it as El Nino factor. That will be set up in the month of July or August. Maybe this season, therefore, there will be periodically drought situation in many parts of the country. This is another challenge. So in agriculture, you will always find that such challenges emerge every year in a different form. Pathology is a permanent challenge. There are pathogens which are the permanent challenge, which as a scientist we are tackling. But some of them are not to be done by a scientist alone. Maybe we will have to do it with large amount of policy interventions in between. This is what is happening today. As far as plant pathology is concerned, which the conference which you have organized in a very nice way, I would tell you in a very brief way. You see, we had, as a student, I was, I think, a student of pathology nearly 50 years ago. Then I studied a pathology. I compare what I studied then, has it any relevance today? It's very important. They told me that time that if you have to manage disease, there are 4 major principles of management. One is they call it as quarantine. Secondly, they call it as sanitation, that is clean cultivation. Third, they call it as chemical management, you use pesticide. And the third, they call it as, fourth is called as host resistance. Now, these are the principles. If you look any pathology book, these principles still exist. Now, the question is, if I use those again, the methods of applications of those principles today, they are not applicable. हमारे यहाँ जैसे एक we had a pathology, the farmers, they normally come to a pathologist as a doctor, that please see, I have certain diseases, can you recommend something? And हमारे pathologist भी ऐसा सोचते हैं कि I am a doctor, pathology doctor. Actually, he is not. He is a scientist first and doctor later. तो एक pathologist एक farmers लेके आया, farmers in my university, Parbhani. He brought some samples of the foliage, leaves. और वो हमारे पैथोलॉजिस्ट के पास आया. This is a story of about 40 years ago. क्योंकि उस टाइम डायग्नोस्टिक मेथड ऐसी थी कि either you look into microscope or you see the sample and suggest that the symptoms are like this. तो उन्होंने देखा तो he says, oh, this is a bacterial blight of cotton. एंड आप ऐसा करिए कि इसमें कॉपर ऑक्सीक्लोराइड है 2%, 0.2%, 2%, आप स्प्रे कर दीजिए. That farmer was very intelligent. He said, sir, this is okay. But this leaf जो है, ये cotton का नहीं, ये grape का leaf है. So you can imagine that you have, as a pathologist, the very first thing is you have to be a very good botanist to understand what is a crop. Otherwise क्या होता है कि if you don't know crop and you know disease, has no meaning. So that's why I always say that these professions, like say if it is pathology or all that, you have to study the plant first and then come out. Now regarding the diagnostics, you will see, this is very interesting. That earlier we used to see only, as I said, in the basis of symptoms which we already get and then learn that okay. But today it has undergone several series of changes. Now we are using, as Dr. Chahal was telling, we are using now AI methodology. We are using now sensors to diagnose. We have now small equipments which you take to the field, take a photograph, and directly on your mobile you get information that this is the disease. Technology has changed. So same thing, diagnostic and monitoring, they are all changing. Second thing is which we always say that we have chemical management. Now we have already seen that what is happening by using of chemicals on an excess way. Green revolution has taught us so many things, but at the same time it has also taught us that kindly do not use more of only chemical, because it has lots of side effects. And naturally, as Dr. Swaminathan always used to say, what are the dangers of green revolution? Swaminathan Devi always said that we should look into evergreen revolution, not a green revolution. Because green revolution, there what we achieved in a short span of time by using pesticides for management or by using the fertilizers by management, this should not be a last long sort of a profession. It must change over a period of time. And that's how he said that if we want to achieve evergreen revolution, it must be sustainable management of anything. And that is how the second aspect, chemical control, now we have slowly shifted to microbes. Now there are lots of papers which I see every conference, they are on microbiology. They are more on microbes or what called as biopesticides or pesticides which are microorganism originated or plant originated. And that's how they are able to now replace large number of chemical management. Now when the third aspect or fourth aspect that which we talk about is breeding. We developed resistant varieties of wheat by traditional breeding by over a period of almost 7 years, 8 years. One variety to develop it used to take 7 years. Today, the technology for developing varieties has also changed. Either we can use what we call it as a speed breeding technology, we can use GM technology, or we can use genetic engineering technologies, gene editing technologies to develop. Probably you must have already read that ICAR has released 2 rice varieties using gene editing. Now gene editing, some part of gene editing has already been approved or permitted. It doesn't normally go to the regulatory way. It has been already accepted that SDN1 and SDN2 technology is accepted. And that's how now every laboratory you will find or every institution must think that if I have to develop, because time is fast, climate change is affecting the damage. So you have to also go fast in technology to really go fast development of varieties. And that is how gene editing technology has come. So resistant varieties remain our backbone of principle. But the way now we develop resistant varieties will change. So this is how all the 4 principles of management remains the same, whether it is sanitation, quarantine, whether it is the chemical control, or now we have also to see that what are the chemicals which do not cause any side effect. So those safe chemical technologies, what we normally use is the green technologies, they are also coming fast. So all these changes are coming over a period of time, and I am certainly sure that this conference which you are already organizing, for which I congratulate the organizers, they will discuss some of those issues, and then probably we can have those proceedings where the students will benefit. Because I am only worried what the next generation of scientists or technology developers are the students. And they must take interest in science. Unless you take interest in science, I don't think it will be possible. So I think we have now certain changes in our program. There will be some, I was told that Honorable Minister will also be arriving very soon. So maybe we'll break for some time. Maybe he arrives or yeah. So because there is already a special lecture by me, which is related to more on sensor technologies and AI, which I will discuss it little later. But I wish to again thank the organizer for giving me this opportunity to interact with you in the inaugural session. Thank you very much.
Thank you very much, sir, for enlightening all of us. Deputy CM, sir, is on the way and he will reach in a short while from now. I request all of you to rise on your seats when Deputy CM, sir, arrives. In the meantime, God has granted a wish, which we also are very happy to call again Dr. Lalit Mahatma, sir, President IMPP, to address on the behalf of ISMPP. ये हमारा सौभाग्य है कि हमें पुनः सुनने का उन्हें अवसर मिल रहा है. Most welcome, sir. And this is his presidential address on the behalf of IMPP. Thank you very much. ISMPP, sorry, ISMPP.
A very good afternoon to all of you. Delegate at the dais, delegate of the dais, august member of Indian Society of Mycology and Plant Pathology. At the outset, I would like to express my sincere thanks to the member of Indian Society of Mycology and Plant Pathology for nominating me as a president of this high-valued, esteemed society. In fact, we all know that there are only two societies in this country who's taking care of different scientific activity of plant pathology. And ISMPP is one of them. Dear friends, as a president, I always try to keep the values of society at the high and always try to build a very nice team. I believe in the philosophy that लगे चोट पाँव में, I'm sorry, ये झेलना पड़ेगा आपको जब तक मैं हूं. लगे चोट पाँव में तो घाव बनते हैं, मुसीबतों से जीवन में ठहराव बनते हैं. अकेले चलने से कुछ भी नहीं मिलता, अकेले भटकने से कुछ भी नहीं मिलता. मिलते हैं लोग कहीं एक साथ तो गांव बनते हैं, मिलते हैं कहीं लोग एक साथ तो गांव बनते हैं. And we try to, make this society as a village, as a country, as a world for all of us. So I'm very much thankful. Among the different members who have contributed a lot and helped me for doing the activity of this society, few of them are sitting. Most notably, I would like to express my sincere thanks to Dr. S.S. Chahal, sir, who is a father figure for all of us. And as a father carry his child when they are young and going first time to the school by holding his finger, he has taken us like this. लोग मुझे बोलते हैं, मेरा हाथ मुझे बोलता है कि आपकी इस अंगुली से हम जलते हैं, क्योंकि आपकी इस अंगुली को चहल साहब ने पकड़ रखा है. Thank you very much, sir, for holding us. I am very much thankful to our CD Mai Sahab. Many stalwarts are sitting there here for the blessings. Every time, whenever there was any program or there is any planning, we by default consider that the blessings of Honorable Sidi Mai Sahab will be with us, and it had been with us, and it will be with us for another 30 years or more. He is very young. He is younger than me in all the activities, except that he doesn't color the hair and I color the hair. Thank you very much, sir. In fact, during my first presidential address, if you remember, I tried to speak about the biopesticide, especially the trichoderma. And in this trichoderma, there are so many issues. One of the issues, what I wanted to highlight was this, that are we seriously promoting trichoderma as a biocontrol agent or not? Because when we talk about the biocontrol agent and use of the trichoderma, again, Mukhopadhyay Sahab is sitting here. I'm too small to speak anything about the trichoderma in front of him. And when I was delivering my presidential address, he was chairman of that session. Dear friends, the trichoderma is doing very well in the lab. The trichoderma is doing very well in the control conditions. But we are not getting the due result which we are expecting from the trichoderma. And there are few things which I try to discuss. I will not repeat it. And the most important point I raised in my first presidential address was this, that certainly we have to relocate the doses, what we are recommending to the farmers for the trichoderma especially. What is the criteria behind this, deciding the doses? We have to look into this. And I discussed that if you are going to use 10 kilogram trichoderma per hectare, and the trichoderma is having 5 into 10 raised to power 7 CFU per gram, if you will make a very meticulous and accurate calculation by calculating the area which is being covered by this, you will find that only the 12 spores of trichoderma are coming per gram. At the same time, if you will see that the inoculum, generally what we call as the inoculum potential of the pathogen, and we calculate the inoculum potential of the pathogen, we generally come across this. Now, of course, it varied from pathogen to pathogen and its pathogenicity factors. But you'll find that around 3,000 CFU of the pathogen would be there to incite the disease in that particular conditions. I'm especially talking about the soil-borne diseases. So this was the issue as a president I wanted to address. Here also today I'm coming with a very important topic which has not been paid due attention. And I hope that after my presidential address, all the scientists will pay due attention to this important topic, that is seed transmission of begomovirus, unresolved mystery requiring rigorous scientific evidence and greater research attention. Dear friends, we know, as Dr. Salini also in her address, Sidi Mai Sahab also in his address, and Chahal Sahab also in address, somehow said that the seed is the most important vehicle to take the pathogen from one season to another season, or to take one country to the another country. So the seed is the most important vehicle to take the pathogen from one country to another country and carry forward the pathogen from one season to another season. We have seen that the diseases which is transmitted through the seeds are transmitting from one country to another country very rapidly. That's why we are paying due attention towards the seed transmission or seed-borne diseases. In fact, when we talk about the seed-borne disease and seed transmission, then we come across with the quarantine law also. There are robust quarantine law which prevent the spread of many diseases from one place to another place. It may be the indigenous or it may be the international quarantine that everybody knows about it. I'm talking about the begomoviruses, dear friends. If you talk about the general seed transmission of the virus, bean common mosaic virus is the first virus which was known as a seed transmitted long back in 1927 around. After that, we have found so many viruses are transmitting through the seeds. But when we talk about the begomovirus, before 2010, we always used to claim that the begomoviruses, they are the Gemini virus, they are single-stranded DNA containing viruses, they are transmitting from seed to other, or they are transmitting from one plant to another plant by the exclusive whitefly that is Memitia tabeci in a semi-persistent manner. In 2008, when I was visiting the field of my university, Navsari Agriculture University, I observed that certain plants are showing the yellow color canopy leaf, and the pods are also of the yellow color. When the pods are also of the yellow color, then out of curiosity, what I did, I opened the pod and tried to observe whether the seeds are also having yellow color or not. The seeds are green color or yellow color. When I opened that pod, I observed that certainly the tinge of yellow color is also there on the pod. And immediately the question came in my mind that so far we have said that the begomoviruses are not transmitted through the seeds. But if the color of seed coat has turned yellow, certainly there is some activity of the viruses in the seed coat also. I took the sample to my lab. I asked my MSc student to do the PCR of this by using the degenerate primer specific for the begomoviruses. He did the PCR of the seeds, and to surprise of all of you, we observed that the virus is present in the seeds. I would like to repeat the things which is very important. I feel that the virus is present in the seeds. This was the first observations we made in 2008, that the virus is present in the seed. But since before that nobody has said, and we are so small scientists, still I am so small when we talk about the science and knowledge of the people, it's the ocean, I'm a drop of it. What I did, I opened the seeds and dissected it. I separated the seed coat, I separated the cotyledon, I separated the embryonic axis. I observed that the virus is there in the seed coat, the virus is there in the embryonic, the virus is there in the cotyledon, but the virus is not there in the embryonic axis. I went for the storage of the seed, because see what happened, that if you have harvested the seeds and have observed the virus in that, means certainly there may be the chances that virus may survive for some time in the tender or the young leaves. But when it grows, it may lose viability, or the virus may not survive into this. This is one of the reasons that before 2008, we never said that the begomoviruses are seed transmitted. Still, I'm not using the word seed transmitted, but begomoviruses are present in the seeds. In storage also, when we saw after 1 year, we observed that the virus is present in the seeds. Of course, the second question comes here, that if the virus is present in the seeds, and the PCR is a technology where whether the virus is surviving or not, it will detect it. So to confirm this, whether the virus is surviving or not, we have for the exenic culture, through the tissue culture, we developed the callus from the cotyledon, and when we developed the callus from the cotyledon, we observed that the virus is present in the developed callus, means the virus is surviving. It has multiplied in the callus, what we have developed. I took around 6 to 7 years to do this work, because when you are saying something new to the scientific fraternity, you have to double sure or triple sure on it. In 2010, the boy submitted his thesis. So it was documented in the thesis that this is happening. In 2015, I have a deep love with Indian Society of Mycology and Plant Pathology. And you can have only one wife and many girlfriends, but I am a person who have only one wife and one girlfriend, that is Indian Society of Mycology and Plant Pathology. Of course, my wife is sitting here. So, so I thought that, okay, I will present this research finding in this Indian Society of Mycology and Plant Pathology somewhere. And in 2015, I presented this in the month of February at Tamil Nadu Agriculture University, Madurai. It was the first presentation where we said that the begomoviruses are present in the seeds. But I wanted to maintain a legitimate distance from the word, that whether it is a seed-borne or seed transmitted. Because the moment I use the word it is a seed-borne, or the moment I use the word it is a seed transmission or seed transmitted, the eyes of Dr. Jamil Akhtar is here, eyes of the quarantine people will be big. They will think over it. Many loss will be there to the nation. Many people or many consignment may be rejected on the basis of this, that the virus is present into this. So what I did, I presented here in that ISMPP conference as a role of seed in the epidemiology of seed-borne Mungbean yellow mosaic virus. I presented that paper, sir, on 12 to 15 February 2015, and from the Korea, from the South Korea especially. On 21st February 2015, a research paper came in which they said that sweet potato leaf curl virus, which is a Begamo virus, is transmitted through the seeds. The word used was the seed transmitted, isn't it? So they reported in 2015 that the sweet potato leaf curl virus is a seed transmitted. Here I would like to clear to the August Society members that generally in our place, we go for the vegetative propagation of sugar beet, sweet potato, sorry. But sweet potato seeds are also produced for the breeding purpose and other purposes also. In some places, it is propagated through the seeds also. So from South Korea, that was the report. Dear friends, when there was a report from the South Korea, then again there was a report from Tamil Nadu Agriculture University that the Mungbean yellow mosaic virus in the black gram is a seed transmitted, isn't it? So of course, it's a seed borne. So here two words comes. One is a seed transmitted and another one is a seed borne. And we all know that the father of seed pathology in the world is Dr. Paul Nirgard. He has written classical book with the 2 volumes. In that also, if you will go through the different chapters and different part of the Paul Nirgard seed pathology, both the volume, you'll find that the many places seed-borne and seed-transmitted word is interchangeably used. So anywhere it is seed-borne written, means the virus is carried, or any pathogen, whether it is a bacteria, fungi, or a nematode even, is carried through the seeds. And it is a seed transmitted, confirm it is carried from the seed. But somewhere they have specified in their book also that there is a difference. If the virus is present in the seed, it is a seed-borne. But if the virus is carried from the seed, it is a seed transmitted. But still this confusion is there in front of all the scientific society. And many people who are reporting seed transmission is writing seed-borne, and vice versa it is happening in the case of this. Anyway, dear friends, so like this, in the entire world, if we talk, I can say that there are 3 working groups in this entire world, which is saying the Begamo virus, its presence, its transmission, or its seed-borne nature. When we discuss about the seed-borne nature and seed transmission nature, then again the key that the Korean group only, they have reported that the tomato leaf curl Israel virus is also seed transmitted in the tomato. They prepared a clone of it, and through agro inoculations, they have inoculated the plant and observed that this virus can be transmitted from plant to seeds, like artificially inoculation by using the agro inoculations, then transfer in the soybean plant, tobacco plant, tomato plant, and different plant, and then it's a subsequent transmission into the seeds. And that again indicate that certainly the channel between the planted seed is robust. Dear friends, many people after that has reviewed these things and has made the things. But around 2017 and 18, Rosas and their friends from the Spain first time said that the Begamo viruses are present in the seeds, but it is not the seed transmitted. The Begamo viruses are present in the seeds, but it is not seed transmitted. When the Begamo viruses are present in the seeds and it is not seed transmitted, the first study was on the tobacco. And tobacco, when we know about all these things, that when we do this, generally we use the Nicotiana benthamiana and Nicotiana glutinosa for the different biotechnological activities, not the other tobacco. So they observed this, that in Nicotiana benthamiana, the virus is not transmitting from seed to seedling. After that, there was a confusion again. After that, there was a confusion again because it has some quarantine issue and all. From the Spain and from the US, where the import and export of these material was high, they started working on this, especially two viruses. One is a tomato leaf curl virus, and the another one is a, just one. Sorry. So they started working on this, and the report started coming like this. The sweet potato leaf curl virus is not seed transmitted. The sweet potato leaf curl virus is not seed transmitted. And when we talk about the earlier research, what the researcher has done and the observations has done in a very high impact papers from the British Society of Plant Pathology and American Society of Plant Pathology even. जब आप कहोगे मैं खत्म कर दूंगा. Thank you. So dear friends, they observed that the virus is not transmitted. And when the earlier transmission rate was observed, it was more than 30%, 40%, 90%. We know that even, Jamil sir will be agree with this, that even 0.01% seed transmission is observed. Quarantine law comes into this. It is sufficient to cause the epidemic in any of the area if the congenial environmental conditions and vector to transmit that particular virus is available in that area. Here, it is 30%, 60%, 80% even in some cases. The seed transmission is there. Dear friends, when these things was challenged, so they have done research from the Spain and from the US, from the China. So there are 3 centers of seed transmissions or presence of virus in the seeds. We never said that it is a seed transmitted. We always said it is a presence of virus in the seeds. But from the Tamil Nadu Agriculture University and from the Korea, it has said that they are seed-borne or seed transmitted viruses. Then there was a question on it from the Spain that they are not. And it is not based on a small observations. Rather, it was based on the observations where more than 300 seeds were taken for the observations. There were more than 3,000 to 6,000 seeds were tested and they found it. So in 2015, the most importantly, we thought that if it is not seed transmitted or it is present in the seed and not moving from the seed to seedling, then why it is coming in the seeds? If you believe in the God doesn't create anything unnecessarily. He doesn't give anything unnecessarily. And if you believe in the evolution theory, then in which way the evolution of begomoviruses took place, that it has come to the seed, and from seeds it has facilitated to move from one season to another season or from one area to the another area. When we did all the observation, then we came to know that especially I'm talking about the mung bean yellow mosaic virus, that the mung bean yellow mosaic virus is present in the cotyledon. We observed it that the germination of mung bean yellow mosaic, germination of the mung bean is apogel type germination. And when the germination of mung bean is apogel type, then the seed coat rapture the crust of soil and come out. When seed coat come out, it imbibe water and become the succulent. Here, no cell division takes place, but the complex food is converted into the simple food to provide the nutrient to the developing seedling. When the complex food is converted into the liquid simple food glucose for providing the nutrient to the developing seedling, it becomes succulent and attract whitefly and cause the disease or spread the disease. So thank you very much to all of you for giving me a chance to deliver my presidential address. Thank you very much.
Thank you very much, sir. And now that moment has arrived. Deputy CM sir is here. He can be here any moment. कृपया अपने स्थान पर खड़े होकर करतल ध्वनि से स्वागत करेंगे आप। माननीय उप मुख्यमंत्री केशव प्रसाद मौर्य जी हमारे बीच आ चुके हैं। जोरदार तालियों के साथ स्वागत करें माननीय उप मुख्यमंत्री श्री केशव प्रसाद मौर्य जी का। आपकी करतल ध्वनियां कम पड़ रही हैं। पुनः और पुनः करतल ध्वनि से एमिटी में हार्दिक अभिनंदन है हमारे माननीय उप मुख्यमंत्री श्री केशव प्रसाद मौर्य जी का। एंड आई वुड आल्सो लाइक टू acknowledge the online presence of Honorable Founder President Dr. Ashok K. Chauhan sir and Honorable Dr. Asim Chauhan sir, Chairman, Lucknow Campus. देवियों और सज्जनों, मुझे अत्यंत गर्व, हर्ष और गहन सम्मान के साथ स्वागत करने का सौभाग्य प्राप्त हो रहा है हमारे मुख्य अतिथि माननीय उप मुख्यमंत्री उत्तर प्रदेश श्री केशव प्रसाद मौर्य जी का। आप एक दूरदर्शी नेता, जन सेवा के प्रति समर्पित व्यक्तित्व और प्रगतिशील उत्तर प्रदेश के निर्माण में अग्रणी शक्ति हैं। आपका नेतृत्व विकास, सुशासन और जन कल्याण का एक सशक्त उदाहरण है जो समाज के हर वर्ग को सशक्त बनाने की दिशा में निरंतर कार्यरत है। आज आपकी गरिमामय उपस्थिति न केवल इस आयोजन की शोभा बढ़ा रही है बल्कि हम सभी के लिए प्रेरणा का स्रोत भी है। सर, हम आपके आगमन से अत्यंत गौरवान्वित हैं। एमिटी विश्वविद्यालय लखनऊ कैंपस उत्तर प्रदेश की ओर से मैं आपका हार्दिक स्वागत और अभिनंदन करता हूं। सम्मान केवल एक परंपरा नहीं है, यह प्रेरणा, समर्पण और उत्कृष्टता का उत्सव है। अब हम सम्मान करेंगे अपने मुख्य अतिथि माननीय केशव प्रसाद मौर्य जी का। मैं सादर आमंत्रित करता हूं डॉक्टर अनिल वशिष्ठ, माननीय प्रो वाइस चांसलर, एमिटी यूनिवर्सिटी उत्तर प्रदेश। And before that, we have the lighting of the lamp ceremony.
या कुन्तेन्दु तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। शुक्लां ब्रह्म विचारसार परमां आद्यां जगद्व्यापिनीं वीणां पुस्तकधारिणीमभयदा।
असतो मां सद्गमय, तमसो मां ज्योतिर्गमय, मृत्योर्मामृतर्गमय। इस दीप की ज्योत हमें असत्य से सत्य की ओर, अंधकार से प्रकाश की ओर और मृत्यु से अमरता की ओर ले चले। जिस प्रकार यह दीपक चारों ओर प्रकाश फैलाता है, उसी प्रकार यह सम्मेलन भी हमारे विचारों को प्रकाशित करे, ज्ञान को विस्तार दे और हमें एक सशक्त एवं सतत भविष्य की दिशा में प्रेरित करे। सम्मान केवल एक परंपरा नहीं है, यह प्रेरणा, समर्पण और उत्कृष्टता का उत्सव है। अब मैं निवेदन करूंगा Professor Dr. Anil Vashisht, Pro Vice Chancellor, उत्तर प्रदेश लखनऊ कैंपस एमिटी यूनिवर्सिटी से, और प्रोफेसर डॉक्टर शालिनी बिसेन जी से कि माननीय उप मुख्यमंत्री जी का प्लांटर देकर सम्मान करें। साथ में अंग वस्त्र से भी सम्मानित करते हुए प्रोफेसर अनिल वशिष्ठ, कृपया करतल ध्वनि से आप सभी माननीय उप मुख्यमंत्री जी का स्वागत करें। And also felicitating ISMP की तरफ से डॉक्टर पोखर रावल, सेक्रेटरी ISMP, डॉक्टर चहल, माननीय उप मुख्यमंत्री जी का विशिष्ट सम्मान करते हुए now an abstract book will be released, and before the release of abstract book, an AV will be played. Kindly play the AV of abstract book. ISMPP 5th International Conference on Pathogens, Plant Health and Food Security, recent advances for climate resilient agriculture and landscape. Released by the Honorable Deputy CM Monaji and the dignitaries on this stage. It's now my privilege to invite a visionary leader whose commitment to excellence continues to inspire generations. Honorable Dr. Asim Chauhan, sir, a dynamic torchbearer of innovation and excellence who continues to redefine the horizons of global education. Sir, we request you to kindly share your words of wisdom.
सबसे पहले तो मैं माननीय श्री केशव प्रसाद मौर्य जी को, ऑनरेबल डेप्यूटी चीफ मिनिस्टर उत्तर प्रदेश का अभिनंदन करता हूं। उनको एमिटी यूनिवर्सिटी लखनऊ कैंपस में स्वागत करता हूं। सर, आपके आने से हम सबका प्रस्थान बढ़ा है, ना तो सिर्फ उनका जो साइंटिस्ट और स्टूडेंट एमिटी यूनिवर्सिटी लखनऊ कैंपस में बैठे हैं, लेकिन वो सब साइंटिस्ट, वो सब स्टूडेंट जो पूरे भारत में और कई देशों में ऑनलाइन लाइव स्ट्रीम के द्वारा आपको सुनने के लिए वेट कर रहे हैं। आप इतने व्यस्त होते हुए भी आप आए, आपका बहुत बहुत धन्यवाद। हम आपको हृदय से आभार प्रकट करते हैं कि आपने समय निकाल के हमारे पास आ जाए। श्री केशव मौर्य जी ने पूरी जिंदगी समाज सेवा में लगाई है और ग्रामीण क्षेत्र में भी उन्होंने हमेशा सोचा है कि जो हमारे किसान भाई बहन है उनकी प्रगति कैसे हो, उनकी उठान कैसे हो। तो या तो रूरल इंफ्रास्ट्रक्चर हो, जहाँ पे आपने बहुत रूरल रोड्स और ब्रिजेस बनवाए हैं, फूड प्रोसेसिंग हो, माइक्रो इरिगेशन हो, फार्मर इनकम को बढ़ाने के लिए वैल्यू एडिशन हो अलग अलग उत्तर प्रदेश के एरियाज में, और अब लेटली ड्रोन और एआई का इम्प्लीमेंटेशन ग्रामीण क्षेत्र में कैसे हो, ये सब मौर्य जी के लीडरशिप में डेवलपमेंट हो रहे हैं। और मैं सोच रहा था कि जो हमारे आदरणीय प्रधानमंत्री जी का एक विजन है विकसित भारत का, अगर वो पूरा करना है तो योगी आदित्यनाथ जी और केशव मौर्य जी जो एग्रीकल्चर में काम कर रहे हैं उत्तर प्रदेश में, उसका बहुत बड़ा हाथ है। हमारे ऑनरेबल फाउंडर प्रेसिडेंट लाइन पे हैं, उन्होंने कहा कि पहले मैं मौर्य जी के शब्द सुनूंगा, उसके बाद मैं अपने कमेंट करूंगा। तो डॉक्टर अशोक चौहान जी, जो हम सबके फाउंडर प्रेसिडेंट ही नहीं, हम सबके इंस्पिरेशन हैं, वो भी शुरू से ही ऑनलाइन थे सुनने के लिए कि हमारे जो ISMPP के कॉलीग्स हैं वो क्या बताने के लिए आए हैं। लेकिन खास गौर जी कि मौर्य जी आए हैं तो उनको सुनने के लिए कि उनका विजन क्या है कि हमारे स्टेट का, हमारे राज्य का, हमारे देश का कैसे उठान हो। I would like to congratulate all organizers, Shalini Singh Vishenji from the Amity Food and Agriculture Foundation. You are a great scientist. You have a number of very large projects in the area of agriculture, and आपने लखनऊ में एक मशरूम सेंटर ऑफ एक्सीलेंस इनॉग्रेट करा था करीब 2 साल पहले. So I congratulate you for being from the Amity side, the forefront, taking the guidance of our very dynamic Pro Vice Chancellor, Dr. Anil Vishisht, and the entire team. And I must congratulate this wonderful society, the Indian Society of Mycology and Plant Pathology, ISMPP. उसमें डॉक्टर ललित महात्मा जो प्रेसिडेंट हैं उनके हमने शब्द सुने, डॉक्टर पोखर रावल जी के हमने शब्द सुने, और जो जो ऑर्गेनाइजिंग कमेटी में इन्वॉल्व है, संजय जी हैं और भी हैं, आई कंग्रेचुलेट यू। आपने जो आज पब्लिकेशन लॉन्च करवाया है, लेकिन आप जो रिसर्च कर रहे हैं कि हमारे कंट्री में सस्टेनेबल एग्रीकल्चर कैसे हो, हमारे क्लाइमेट रेजिलियंस कैसे हो, उसकी बड़ी इंपॉर्टेंस है, एंड आई एम श्योर दैट वी विल लर्न अ लॉट फ्रॉम दिस. We also had 3 very eminent guests of honors. Dr. C.D. Mai, you have spoken wonderfully. Dr. D.R. Singh, the Vice Chancellor of Bihar Agriculture University, and Dr. S.S. Chahal, who is a diggage, if I can call, in this area of pathogens and agriculture and plant pathology, former Vice Chancellor of Udaipur, MPUAT, who we have known for many years. So, Professor Chahal, good to see you on the platform today at Amity Lucknow campus. Amity is committed to agriculture research and food security. अभी मैं सोच रहा था कि पिछले 2 हफ्तों में मेरे पास 4 हमारी यूनिवर्सिटी से मैसेज आया कि वहाँ पे जो हमारे केटरिंग मेस में जो हमारे केटरर्स हैं उनको मेन्यू बदलने पड़ रहा है, कि जो नॉर्मल मेन्यू है वो नहीं बना पा रहे, क्योंकि जो स्ट्रेट ऑफ हॉर्मूज में जो ईरान US इसराइल की वॉर चल रही है, उसकी वजह से जो गैस की शॉर्टेज है, उसकी वजह से जो नॉर्मल खाना बन रहा है वो नहीं बन पा रहा. तो एक बच्चे ने कहा, सर कर्नाटका में डोसा नहीं मिल पा रहा मुझे. तो मुझे लगा कि हमारी फूड सिक्योरिटी कितनी नाजुक है कि वहाँ पे तो सप्लाई चेन इशू की वजह से हमारे मेन्यू चेंज हो रहे हैं, लेकिन कल कुछ भी हो सकता है. अगर क्लाइमेट चेंज ज़्यादा इफेक्ट हो, अगर पैथोजेंस का कोई इम्पैक्ट हो, तो अगर हमें एक इंडिपेंडेंट कंट्री बन के आगे बढ़ना है, तो सस्टेनेबल एग्रीकल्चर और फूड सिक्योरिटी की बहुत अहमियत है. और इस कॉन्फ्रेंस में जो आइडियाज़ आएंगे, यहाँ से जो रिसर्च निकलेगी, उसमें जो उसका जो इम्पैक्ट होगा बड़ा डीप होगा, लॉन्ग थिंकिंग होगा. एमिटी यूनिवर्सिटी में एग्रीकल्चर में बहुत काम हो रहा है. पिछले 1-2 साल में 5,000 पब्लिकेशन एग्रीकल्चर के एरिया में हमारे साइंटिस्ट ने छापे हैं. 2,800 पेटेंट जो हमारे साइंटिस्ट ने फाइल करे हैं, उसमें से 150 पेटेंट एग्रीकल्चर के एरिया में है, जिनमें से 50 पेटेंट ग्रांट भी हो चुके हैं. इस वक्त 50 नेशनल इंपॉर्टेंस के एग्रीकल्चर में फंडेड प्रोजेक्ट एमिटी यूनिवर्सिटीज में है, अलग अलग कैंपसेस में, और बहुत सारे टेक्नोलॉजीज हमने टेक्नोलॉजी ट्रांसफर करे हैं इंडस्ट्री में. तो ये जो कॉन्फ्रेंस है बड़ी इंपॉर्टेंट है, और आई कंग्रेचुलेट डॉक्टर महात्मा, द प्रेसिडेंट ऑफ ISMPP. वी आर ग्रेटफुल फॉर योर कोलैबोरेशन. We commit that we at Amity will keep doing work with you. And Shri Keshav Prasad Maurya ji, we are deeply grateful to you. आप आए, आपके आने से ही जो केमिस्ट्री, जो एनर्जी हमारे कैंपस में बढ़ी है, हम अपने मिशन को, हम अपने गोल्स को, हम एमिटी के जो हमने अपने सेट करे हैं गोल्स, उनको आज बढ़ाएंगे, क्योंकि आपने आज आके हमारी मोटिवेशन इतनी बढ़ाई है, इंस्पायर करा है. इन्हीं वर्ड्स के साथ बहुत बहुत धन्यवाद. थैंक यू.
Thank you very much, Honorable Chairman Sir, for your word of wisdom. माननीय मुख्यमंत्री जी, जैसा कि हमारे चेयरमैन सर ने कहा, हमारे फाउंडर प्रेसिडेंट सर आपको सुनना चाहते हैं. आपके उस विकसित भारत की माननीय प्रधानमंत्री जी की परिकल्पना को साकार होते हुए देखना चाहते हैं और कदम से कदम साथ मिलाकर एमिटी इसमें आपके साथ है। और जी हाँ, अब वो क्षण आ गया है जिसका हम सभी बेसब्री से इंतजार कर रहे हैं। मैं सादर आमंत्रित करता हूं माननीय उप मुख्यमंत्री उत्तर प्रदेश श्री केशव प्रसाद मौर्य जी को, जो अपने प्रेरणादायक विचारों से हमारा मार्गदर्शन करेंगे।
सबसे पहले तो मैं अमेठी यूनिवर्सिटी परिवार और जो इस कार्यक्रम के साथ जुड़े हुए कई संस्थाओं के नाम है, मैं सबके नाम नहीं ले रहा हूं, उन सभी का धन्यवाद करता हूं कि उन्होंने इस प्रकार के आयोजन की परिकल्पना किया, जिसके आधार पर भारत का वर्तमान भी और भारत का भविष्य भी, उत्तर प्रदेश का वर्तमान भी और उत्तर प्रदेश का भविष्य भी मजबूती से आगे बढ़ सकता है। और आज के इस कार्यक्रम में हमारे साथ मंच में प्रोफेसर डॉक्टर शालिनी सिंह बिसेन जी, जो ऑर्गेनाइजिंग सेक्रेटरी के रूप में, डायरेक्टर के रूप में जिम्मेदारी निर्वाह कर रही हैं। हमारे लखनऊ अमेठी यूनिवर्सिटी के वाइस चांसलर प्रोफेसर डॉक्टर अनिल वशिष्ठ जी, हमारे प्रोफेसर डॉक्टर पोखर रावल जी, जो सेक्रेटरी ISMMP राजस्थान कॉलेज ऑफ एग्रीकल्चर से यहाँ पर पधारे हैं। मैं उत्तर प्रदेश में आपका स्वागत भी करता हूँ। डॉक्टर ललित महात्मा जी, जो प्रेसिडेंट के रूप में, प्रोफेसर डॉक्टर बलविंदर शुक्ला जी, वाइस चांसलर अमेठी यूनिवर्सिटी उत्तर प्रदेश की दूसरी जो कैंपस है, और डॉक्टर SS चहल जी, जो एमरिटिस प्रेसिडेंट के रूप में आज यहाँ पर सहभाग कर रहे हैं, डॉक्टर DR सिंह जी, जो वाइस चांसलर बिहार एग्रीकल्चर यूनिवर्सिटी से यहाँ पधारे, डॉक्टर CD माई जी, प्रेसिडेंट साउथ एशिया बायोटेक्नोलॉजी सेंटर नई दिल्ली, आपका भी उत्तर प्रदेश में बहुत बहुत स्वागत है। श्री रवीन जी, प्रिंसिपल सेक्रेटरी डिपार्टमेंट ऑफ एग्रीकल्चर, रवीन जी आए? दिखे नहीं, मुझे नाम इसमें लिखा हुआ है। हाँ, हमारे जो चेयरमैन एमिटी यूनिवर्सिटी लखनऊ डॉक्टर असीम चौहान जी, आपके अच्छे विचार में धन्यवाद करता हूँ। डॉक्टर अशोक कुमार चौहान जी, फाउंडर प्रेसिडेंट प्रीतानंद बलदेव एजुकेशन फाउंडेशन अमेठी एजुकेशन ग्रुप के, मैं आपको भी अभी सुनने के लिए उत्सुक हूँ। डॉक्टर जमील अख्तर जी, जो प्रिंसिपल साइंटिस्ट डिवीजन ऑफ प्लांट क्वारंटाइन नेशनल ब्यूरो ऑफ प्लांट जेनेटिक रिसोर्सेज नई दिल्ली, आप यहाँ पर आए, आपका भी मैं बहुत स्वागत करते हुए, और यहाँ जो हमारे बहुत सारे युवा वैज्ञानिक, जो हमारे बहुत सारे रिसर्चर्स, जिन्होंने अलग अलग क्षेत्र में अब तक कीर्तिमान स्थापित किए हैं, मैं उनका सबका अभिनंदन करता हूं। और आप और अच्छे तरीके से सफल हों, मैं इसकी आप सबको बधाई भी देता हूं। आज मुझे आप सबके बीच में जब आने के लिए कहा गया, तो चूंकि विषय ऐसा है, मेरा बैकग्राउंड गांव से जुड़ा हुआ है, मैं एक किसान परिवार में पैदा हुआ हूं, और मैं जो किसान परिवार, और वह छोटी मतलब जोत के किसान, मतलब जिनके पास जमीन कम होती है, ऐसे परिवार से मैं रहा हूं। तो आप सबके सामने जो तमाम सारे विषय होंगे, उसमें मैं जो आप सबको कहना चाह रहा हूं, देखिए हमारे पास ये कह सकते हैं कि देश में सबसे, अगर उत्तर प्रदेश की बात करें, तो उत्तर प्रदेश में जो देश की सबसे उपजाऊ भूमि अगर मैं कहूं कहीं है, तो वो उत्तर प्रदेश में है। अगर विश्व पटल पर हम ध्यान दें, तो भारत हमारा कृषि प्रधान देश है। हमारे पास जो धरती है, वह अनेक प्रकार की उपज देने में सामर्थ्य है। अब उसके लिए रिसर्च आपको करना, जो समस्या आती है कि किसी के पास जमीन तो उपलब्ध है, लेकिन बहुत जगह सिंचाई के साधन नहीं है, कहीं जमीन भी है, सिंचाई के साधन भी हैं, और किसान अच्छी पैदावार भी कर रहा है, और उस पैदावार के बाद जो उसने चाहे वो हरी सब्जियों का उत्पादन करे, चाहे अन्न का उत्पादन करे, तो उसके भंडारण का, उसके कोल्ड चेन का देश की जरूरत है, दुनिया की जरूरत है। हमारा किसान उत्पादन भी कर रहा है, लेकिन उसकी बहुत बड़ी मात्रा में, आप सब रिसर्च करते हैं, आप सबके इस विषय ध्यान में भी होगा कि बहुत बड़ी मात्रा में खेतों में ही फसल बर्बाद हो जाती है। एक उदाहरण के तौर पर मैं आपको बताऊं, आलू का जो किसान होता है, अभी इस वर्ष आलू का उत्पादन थोड़ा ज्यादा हो गया। पिछले वर्ष लोगों ने इस उम्मीद के साथ, किसानों ने अपने जो आलू का भंडारण कोल्ड स्टोरेज में किया जाता है उसका भंडारण किया, और उनको उम्मीद रहती है कि नया आलू जब बाजार में आएगा उससे पहले उसे अच्छी कीमत मिल जाएगी। लेकिन उसे अच्छी कीमत इस बार नहीं मिली है। इसलिए नहीं मिली है कि ज्यादा मात्रा में हमारे पास आलू का उत्पादन हो गया। अब ये एक चुनौती है, ये रिसर्च का विषय है कि अगर आलू का किसान आलू उगाता है, आलू बोता है और उत्पादन ज्यादा हो जाता है, जितनी जरूरत है उससे ज्यादा हो जाता है, हम प्रदेश के बाजार में, देश के बाजार में और विश्व के बाजार में आज के समय में अब कहीं पर भी कोई ऐसी चुनौती नहीं है कि हम दुनिया के, अब जानते हैं आप सब लोग कि भारत ने अभी यूरोपीय यूनियन के साथ भी फ्री ट्रेड डील की है। हम ऐसे कौन से उत्पाद बनाएं कि यूरोप के बाजार में अगर वो जाता है, यूके के बाजार में अगर वो जाता है, तो वहां खरीदने वाला गर्वपूर्वक को भारत से यह सामान बन करके आया है। क्योंकि उसकी तमाम, मुझे ये जानकारी है कि वहां जब तक सर्टिफिकेशन एपेडा आदि के माध्यम से नहीं होता है, तो अगर कोई अन्न का उत्पादन है तो वहां यूरोप वाले नहीं लेते। हम मिडिल ईस्ट में तो बहुत मात्रा में ऐसा भेज पाते हैं, बेच पाते हैं, लेकिन हमारी क्वांटिटी जितनी हो, हमारी क्वालिटी भी उससे बेहतरीन हो, और उस दिशा में आप सबको मुझे लगता है सोचने की आवश्यकता है। आज हम यह भी आप सबके सामने कहेंगे, देश के माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी 2014 से देश की बागडोर संभाले। उनकी प्राथमिकता 4 सेक्टर उन्होंने कहा है। हमारी प्राथमिकता सबसे पहले क्या है? गरीब है। फिर क्या है? किसान यानी अन्नदाता है। फिर क्या है? हमारे युवा हैं। फिर क्या है? तो हमारी मातृशक्ति महिलाएं हैं। इनके सशक्तिकरण के लिए लगातार प्रयास किया जा रहा है। संयोग से जो विभाग मेरे पास है उसमें एक विभाग ग्राम विकास विभाग होता है, और उस विभाग के माध्यम से हम उत्तर प्रदेश में लगभग 1.25 करोड़ गरीब परिवार की महिलाओं को समूह के साथ जोड़े हुए हैं। अब जो समूह से जुड़े हुए परिवार हैं वो कोई ना कोई खाद्य प्रसंस्करण की जो यूनिट है, छोटे छोटे उत्पाद तैयार करते हैं। अब अगर वो उत्पाद तैयार करते हैं, ऐसा उत्पाद होता है जो गांव में तो बिक सकता है, लेकिन अगर उसको लखनऊ में बेचना हो, अगर उसको देश की राजधानी में बेचना हो, देश की आर्थिक राजधानी में बेचना हो, तो वो उस प्रकार से नहीं बिक पाता है, क्योंकि उसकी क्वालिटी ऐसी नहीं है। अभी हम लोग इस क्वालिटी को बहुत अच्छी बनाने की दिशा में काम कर रहे हैं, और आने वाले समय में हमारे समूह की दीदियां भी जो सामान, छोटी सी यूनिट लगा करके अगर कोई सामान का उत्पादन करें, तो वो लाख रुपए से अधिक वर्ष में कमा सकें, ये पहला लक्ष्य हमारा है। ये नहीं कि वो लखपति दीदी बन गई तो उसके बाद उनको आगे नहीं बढ़ना है। वो लखपति दीदी बनने के बाद भी वो करोड़पति बने, यह भी हम लोग लगातार। और बहुत, मैं आपको यह भी बताऊंगा कि बहुत सारी ऐसी भी यूनिटें हैं जो अच्छा उत्पादन करती, और अच्छे उत्पादन करके वो किसानों के द्वारा जो उत्पादित सामान होता है उसको खरीदना, फिर उसकी प्रोसेसिंग करना, उसकी पैकेजिंग करना, और फिर उसकी मार्केटिंग करना, और इसके आधार पर अनेक रोजगार के भी अवसर सृजित होते हैं। हमारे सामने यह भी एक जिम्मेदारी पूर्ण चुनौती है कि हम कैसे अधिक से अधिक रोजगार के अवसर भी सृजित करें। आप सब प्रतिभा से परिपूर्ण हैं। आप सब ने अलग अलग प्रकार से, मैं जानता हूं मुझे आने में देरी हुई, उसके लिए क्षमा भी चाहूंगा। काफी यहां पर चर्चा हुई होगी, लेकिन मैं आप सबको इतना ही कहूंगा, भारतीय जनता पार्टी की डबल इंजन की सरकार है। देश में भी है, प्रदेश में भी है। देश में माननीय प्रधानमंत्री मोदी जी के नेतृत्व में, और उत्तर प्रदेश में हमारे मुख्यमंत्री आदरणीय योगी जी के नेतृत्व में हम लोग लगातार यह प्रयास कर रहे हैं कि उत्तर प्रदेश में बड़े पैमाने पर निवेश भी आवे, और निवेश के लिए कि वातावरण का निर्माण हो। अगर हमारे प्रदेश में बिजली नहीं आएगी तो कोई निवेश करने वाला आएगा क्या? नहीं आएगा। अगर हमारे यहाँ कानून व्यवस्था चुस्त दुरुस्त नहीं होगी, अगर कोई पैसा हमारे प्रदेश में लगावे, उसका पैसा और वह स्वयं भी सुरक्षित नहीं रहेगा तो हमारे यहाँ नहीं आएगा। तो मुझे यह बताते हुए आप सबको हर्ष है कि उत्तर प्रदेश में चाहे सड़क मार्ग हो, चाहे रेल मार्ग हो, चाहे हवाई मार्ग हो, सभी क्षेत्रों में हम लोगों ने देश के अंदर अगर तुलनात्मक दृष्टि से, जो उत्तर प्रदेश पहले बहुत पीछे रहता था, देश के तुलना में जब आप उसका विश्लेषण करेंगे तो आपको ध्यान में आएगा कि आज उत्तर प्रदेश बहुत आगे निकल चुका है और अगर लगातार हम लोग आगे बढ़ने का काम भी कर रहे हैं। हमारी ही सरकार ने एक जनपद एक उत्पाद, मतलब एक जनपद में कोई एक उत्पाद को पहली प्राथमिकता में लिया है, लेकिन आने वाले समय में एक जनपद में कई उत्पाद ऐसे होते हैं जिसकी ब्रांडिंग करते हैं, जिसकी अच्छी पैकेजिंग करते हैं, और आज वो भारत के बाजारों में ही नहीं, उत्तर प्रदेश के ओडीओपी की सामग्री दुनिया के बाजारों में बिक रहा है। मुझे अभी पिछले दिनों उत्तर प्रदेश में निवेश की दृष्टि से जर्मनी जाने का मौका मिला। वहां जर्मनी के जो शॉप हैं, जो दुकानें होती हैं, वहां पर भी हमारे ओडीओपी का सामान बिकता हुआ दिखाई दिया, तो मुझे खुशी हुई। अगर हम अच्छा सामान बनाते हैं तो अच्छा बेचने का भी काम होता है। अभी 24 जनवरी को उत्तर प्रदेश का स्थापना दिवस था, उसमें हमारे देश के गृह मंत्री माननीय श्री अमित शाह जी आए हुए थे। उस दिन एक और योजना को हम लोगों ने लॉन्च किया, एक जनपद एक व्यंजन, यानी कोई एक खाने का सामान, ऐसी ऐसी क्वालिटी के सामान एक एक जिले में बिकते हैं। जैसे कोई लखनऊ में बहुत सारी चीजें, मैं उसका वर्णन नहीं करूंगा, आप बहुत सारी चीजें जानते भी होंगे, वैसे प्रदेश के हमारे 75 जिले हैं, सभी 75 जिलों में कोई ना कोई खाने का ऐसा सामान बनाने का काम किया जाता है जो खाने की इच्छा, जो कोई अगर उस जिले में जाता है तो उस जिले से सोचता है कि ये सामान हम यहाँ से लेके चलें, यहाँ खा के चलें। तो हम जब क्वालिटी को मेंटेन करेंगे, पैकेजिंग को मेंटेन करेंगे, तो एक जिले में ही नहीं, एक प्रदेश में ही नहीं, वो देश में भी बिकेगा। हमारा लक्ष्य जो अभी लगातार बना हुआ है, वह आप सब, एक और चुनौती मैं आप सब यहाँ पर जो बैठे हैं उनके सामने रखूंगा। अभी आपने देखा है 2-3 दिनों से, 4 दिनों से वर्षा हो रही है और पानी के साथ साथ ओले भी गिर रहे हैं। अब ओले भी गिर रहे हैं पानी के साथ साथ, तो ऐसी परिस्थिति में हम किसानों को कैसे, क्या उनकी जो फसल है उसको कैसे बचाने का कर सकते हैं, यह भी रिसर्च का विषय। आप सब सोचिए, यद्यपि जो सामान्य तरीके से जो किसानों के द्वारा कृषि की क्षेत्र में काम करने वालों को करना चाहिए वो करते हैं, लेकिन आप भी इस पे रिसर्च करिए, क्या कर सकते हैं हम। जो हमारा किसान उत्पाद करे, उसकी फसल तैयार है, अचानक पानी बरसा और ओले गिरे और उसकी फसल बर्बाद हो गई। सरकार की एक योजना रहती है कि अगर किसानों को जो भी फसल उन्होंने खड़ी किया, तैयार किया है, अगर इस प्रकार से वर्षा के कारण, इस प्रकार से ओलावृष्टि के कारण वो बर्बाद होती है, तो हम लोग उसका एक परीक्षण करा करके, सर्वे करा करके मुआवजा तो देते हैं। लेकिन मुआवजा देना समस्या का समाधान नहीं है। मुआवजा तो हम देंगे, लेकिन हम ऐसा क्या करें कि हम ऐसे संकटों से फसल की रक्षा कर सकें, किसान का की गई मेहनत बर्बाद ना होने पावे। तो गन्ना की दृष्टि से आप सभी जानते हो, हमारा प्रदेश देश में नंबर 1 का उत्पादन करता है। अब गन्ना उत्पादन में इतना हम आगे हैं। अभी हमारे चौहान साहब चर्चा कर रहे थे कि अभी जो मिडिल ईस्ट में युद्ध चल रहा था, अमेरिका, इजराइल और ईरान का, इसके बीच में आप सब जानते हैं चर्चा का विषय ये बना है। थोड़ी समस्याएं भी आई हैं कि तेल का संकट कोई महसूस किसी को नहीं हुआ, जो गैस का संकट है वह भी महसूस नहीं हुआ, लेकिन वास्तविकता तो यही है कि जब हमारे जहाज जो तेल लेकर के आते हैं, जो गैस लेकर के आते हैं, वो नहीं आने पाएंगे, तो भारत जैसे विशाल देश जहां 150 करोड़ की आबादी, वहां इस प्रकार के संकट खड़े होंगे। तो इस संकट से जो खड़े होंगे, इससे मुक्ति का क्या रास्ता है? हम लोग गन्ने के से एथेनॉल बनाने की प्रक्रिया में आगे बढ़े। अभी कल ही मैं टीवी में देखा हूं, अभी उसका डिटेल मुझे मालूम नहीं है, कि हम परमाणु ऊर्जा बनाने की दिशा में एक लंबी छलांग हमारे देश के वैज्ञानिकों ने लगाने का काम किया है। तो आने वाले समय में हम जब ग्रीन एनर्जी की बात करते हैं, हम अनुकूल पर्यावरण की बात करते हैं, हमारा देश हर क्षेत्र में नंबर 1 का हो, ये हम लक्ष्य लेकर के चलते हैं, हम 2047 जब आजादी के 100 साल पूरे होंगे, यानी आज 2026 में हम हैं, 21 वर्ष बाद जब विकसित भारत बनेगा, और वो बनेगा, उसको बनने से कोई दुनिया की ताकत रोक नहीं सकती है, ये मेरा पूरा विश्वास है। क्योंकि माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के नेतृत्व में जो लक्ष्य निर्धारित किया और हम लोग यही लक्ष्य तक पहुंचे बिना, पथ में पथिक विश्राम कैसा, लक्ष्य है अति दूर दुर्गम, मार्ग भी हम जानते हैं, आप सब जानते हैं, मार्ग भी हम जानते हैं, किंतु पथ के कंटकों को जो भी समस्याएं आएंगी हम सुमन ही मानते हैं, हम फूल ही मानते हैं। यह लक्ष्य लेकर के जीवन के जो उद्देश्य हैं, जिस क्षेत्र में आपने इतना समय का निवेश किया है, और हमारे देश के जो प्रतिभाशाली बच्चे हैं उनका कोई जोड़ नहीं, उनकी कोई टक्कर दुनिया में नहीं ले सकता है। आप हर क्षेत्र में झंडा अपना गाड़ सकते हो, लेकिन आज तकनीकी का समय, अब तकनीक का समय है, तो हम तकनीक का इस्तेमाल किए बगैर आगे नहीं बढ़ सकते। अब जब आगे बढ़ना है तो तकनीकी का इस्तेमाल करते हुए, कृषि के क्षेत्र में एक क्रांति लाते हुए, हम दुग्ध की दृष्टि से, गन्ने की दृष्टि से, अन्न की दृष्टि से देश में सबसे अधिक उत्पादन करते हैं। कई सेक्टर में उत्तर प्रदेश जब भारत के सभी राज्यों की तुलना में हम कम से कम 50 योजनाओं में नंबर 1 स्थान पर हैं, लेकिन हम नंबर 1 पे जब होते हैं तो उससे हम संतुष्ट नहीं होते। क्यों नहीं संतुष्ट होते? कि कई राज्य ऐसे, गिनने के लिए तो राज्य हैं, लेकिन उनकी पॉपुलेशन उत्तर प्रदेश के बराबर नहीं है। उत्तर प्रदेश की आबादी 25 करोड़ से अधिक की है। अब 25 करोड़ से अधिक की आबादी वाले राज्य को देश के अन्य राज्यों की आबादी के साथ भी तुलना करते हुए जब हम नंबर 1 के स्थान पर होंगे, तब हम मानेंगे कि हम माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के लक्ष्य के अनुसार, यानी विकसित भारत का विकसित उत्तर प्रदेश बनाने के काम में सफल हो गए। तो हम इस दिशा में आगे बढ़ना चाहते हैं, और आप सब बहुत अच्छा प्रयास कर रहे हैं। सरकार की मैं तमाम योजनाएं, उनकी चर्चा करूंगा तो बहुत समय आप सबका लगेगा। आप देर से बैठे भी हैं, लेकिन मैं आप सबसे यही कहूंगा, आप तो सब प्रतिभाशाली हमारे युवा वैज्ञानिक हैं और तमाम वरिष्ठ लोगों के मार्गदर्शन में आप काम कर रहे हैं। आप जो भी चीज जानना चाहें, आजकल सब कुछ उत्तर प्रदेश भी तकनीकी का उपयोग करते हुए डिजिटल प्लेटफॉर्म पर हमारी संपूर्ण योजनाएं उपलब्ध, आप उसमें देखिए और क्या अच्छा सुझाव आप दे सकते हैं। हम उत्तर प्रदेश की दृष्टि से, अगर आप प्रदेश की दृष्टि से देंगे वह भी हम आगे बढ़ाएंगे। अगर आप देश की दृष्टि से देंगे, आप भेज सकते हैं डायरेक्ट माननीय प्रधानमंत्री जी को भी, लेकिन अगर आप हमारे पास भेजेंगे तो हम वह माननीय प्रधानमंत्री जी तक पहुंचाएंगे। हम सबका एक ही लक्ष्य है, विकसित भारत। हम सबका एक ही लक्ष्य है, गरीब आदमी अमीर आदमी बने। हम सबका एक ही लक्ष्य है कि हमारा किसान कुछ भी खेत में पैदा करे, वो खेत में बर्बाद ना हो जाए, उसका जो मार्केट वैल्यू है, उस मार्केट वैल्यू के हिसाब से उसके पसीने की बहाने की जो कमाई है उसको समय पर उपलब्ध हो जाए। हमारा सबका एक ही लक्ष्य है कि जब हम अर्थव्यवस्था के आकार को बढ़ाना चाहते हैं, आज हम दुनिया की चौथी अर्थव्यवस्था बने हैं, तो यह आकार कैसे बढ़ेगा? हम अभी मैंने जर्मनी का वर्णन किया, जर्मनी अभी 30 नंबर की अर्थव्यवस्था है, लेकिन उनकी पॉपुलेशन मैं वहां देख रहा था तो 8 करोड़ के लगभग है, और हमारी पॉपुलेशन कितनी है? हम 150 करोड़ की आबादी में अगर चौथी अर्थव्यवस्था है, तो अभी हमारे पास बहुत स्पेस बाकी है, और वह स्पेस पैक करने का काम अगर कोई कर सकता है, कि आप सब वैज्ञानिक कर सकते हो, आप अच्छा मार्गदर्शन दो, आप विश्वास रखो, परिश्रम करके पसीना बहाने वाले लोगों की देश में कमी नहीं है। केवल उनको सही ज्ञान मिलना चाहिए, सही मार्गदर्शन मिलना चाहिए, और जब वो पसीना बहाए और उस पसीने की कीमत उनको जो मिलनी चाहिए वो कीमत मिलने लगेगी। जब कोई सामान को, आप सब जानते हो वैल्यू एडिशन कैसे होगा? वैल्यू एडिशन क्या टमाटर को पैदा करके टमाटर के रूप में बेच देना, आलू को पैदा करके आलू के रूप में बेच देना, उससे वैल्यू एडिशन नहीं होगा। अगर आलू की हम प्रोसेसिंग करके कुछ और बनाने के, पाउडर बनाएंगे, चिप्स बनाएंगे, टमाटर का सब आप जानते हैं, मैं उस पर नहीं जाऊंगा। जब हम इस क्षेत्र में आगे बढ़ेंगे तो हमारी वैल्यू एडिशन होगी, हमारे देश की अर्थव्यवस्था आगे बढ़ेगी। और जो दुनिया की बड़ी अर्थव्यवस्थाएं हैं, आप जानते हैं अमेरिका दुनिया की सबसे बड़ी अर्थव्यवस्था है, दूसरे नंबर पर इस समय चीन है, लेकिन अर्थव्यवस्था का जो आकार है भारत और चीन और अमेरिका के बीच में, उसमें बड़ा गैप है। उस गैप को पैक करने का लक्ष्य लेकर के आप आगे बढ़े। सरकार आपकी है, सरकार आपने बनाई है। हम सबका लक्ष्य, सबका साथ, सबका विकास, सबका विश्वास बनाए रखते हुए आगे बढ़ने का है। और मुझे पूरा विश्वास है कि आज की यह जो कार्यशाला है जिसमें आप सब यहाँ पर सहभाग किए हैं, जो अन्य राज्यों से आए हैं, जो भी हमारे वैज्ञानिक हैं, हमारे सभी वरिष्ठ वाइस चांसलर गण अलग अलग संस्थाओं से, मैं उनका सबका उत्तर प्रदेश की ओर से, यहाँ की 25 करोड़ जनता की ओर से, अपनी सरकार की ओर से हृदय से स्वागत करता हूं। और मुझे इस कार्यक्रम में आपने बुलाया, मैं इसके लिए धन्यवाद करता हूं। आगे इस प्रकार के जो भी कार्यक्रम होंगे, कहीं कोई चीज लगे तो हम अलग से बैठ करके भी सबसे चर्चा करने के लिए उपलब्ध रहेंगे, क्योंकि कल मैं पश्चिम बंगाल में था, वहां चुनाव चल रहा है और चुनाव प्रचार के लिए गया था, इसलिए आज थोड़ा आने में देरी हुई यहाँ पर। इसके लिए क्षमा चाहते हुए आप सबके उज्जवल भविष्य की शुभकामनाएं प्रकट करता हूं। बहुत बहुत धन्यवाद।
धन्यवाद माननीय उप मुख्यमंत्री महोदय। अभी आपने सबका साथ, सबका विश्वास और सबका विकास की जो बात की, इससे मुझे एक वेदोक्त याद आ गई, संग अक्षध्वम संग वदध्वम संभो मनान से जानताम, कि हम साथ साथ चलें, परस्पर एक दूसरे का सहयोग करें और एक दूसरे के साथ मिलकर विकास करें। वो वेदोक्ति अभी भी उतनी ही समीचीन है और विकसित भारत जिसकी परिकल्पना हमारे माननीय प्रधानमंत्री जी ने की है, उसमें हम सभी का सहयोग अपेक्षित है। और कृपया करतल ध्वनि से यह आश्वस्त करें माननीय उप मुख्यमंत्री जी को कि यहां बैठा हुआ हर व्यक्ति विकसित भारत के निर्माण में अपना योगदान देगा। और आपने जिस किसान की बात की और जिस विज्ञान की बात की, किसान और विज्ञान मिलकर देश का उत्थान करेंगे, आपका यह संदेश जन जन तक पहुंचेगा। और जो पंक्तियां आपने कही, मैं पुनः उसको कोट करता हूं आपके अनुबंध से, किंतु पथ के कंटकों को सुमन ही हम मानते हैं। इस रास्ते में कंटक आएंगे, हम उनको सुमन मानते हुए लगातार अपने उस पथ पर आपकी प्रेरणा से आगे बढ़ते रहेंगे। इसके साथ ही मैं पुनः निवेदन करूंगा माननीय प्रोवीसी सर का कि मुख्यमंत्री जी को, उप मुख्यमंत्री जी को स्मृति चिन्ह प्रदान करें, विशिष्ट स्मृति चिन्ह। कृपया करतल ध्वनि से स्वागत करें। And now we seek the blessings of a true architect of modern education, a visionary whose dreams have transformed into institutions of global excellence. May I most respectfully invite Honorable Dr. Ashok K. Chauhan, sir, Founder President, Amity Education Group, to bless this gathering. Sir, over to you.
हम सब बहुत सौभाग्यशाली हैं कि आज श्री केशव प्रसाद मौर्य जी ने अपने अत्यंत बिजी समय से हमारे लिए समय निकाला। मेरे कुछ लोगों से बात हो रही थी, मैंने कहा इतने व्यस्त हैं वो पूरे भारत में, फिर भी वो हमारे यहाँ आए, तो आपने बुलाया? मैंने कहा नहीं। असीम जी ने बुलाया? मैंने कहा नहीं। स्टूडेंट की वजह से आए हैं, वो जो हमारे शोधकर्ता स्टूडेंट हैं, विद्यार्थी हैं, उन्होंने देखा कि जो एमईटी में उत्साह है इनोवेशन के लिए, ऐसे कामों के लिए, और आज जब मैंने उनका बहुत प्रभावशाली वास्तविक रूप का लेक्चर सुना, तो आई थिंक हर एक को जो यहाँ बैठे हुए हैं उससे बहुत प्रोत्साहित होंगे और उनके विचारों के ऊपर आचरण करेंगे। जैसे उन्होंने बिल्कुल ठीक कहा कि हम कृषि प्रधान देश हैं, 140 करोड़ लोग हमारे हैं, उनमें से बहुत बड़ी परसेंटेज में कृषि के लोग हैं। मेरे भी बाबा जी कृषि में थे, तब से मैं जानता हूँ कि कृषि कितना उत्तम क्षेत्र है भारत के लिए। आज का जो प्रोग्राम सवेरे से मैं देख रहा था, तो मैं अनिल वशिष्ठ जी को बहुत बहुत बधाई देता हूँ, और सारे जो ऑर्गेनाइजिंग कमेटी के लोग हैं, इतने बड़े बड़े लोग, इतने रेगुलर इस क्षेत्र में काम करने वाले लोग, पता नहीं कैसे आप लोगों ने उन सबको बुलाया, वो सब आए और बहुत प्रभावशाली लेक्चर दिए। चहल साहब के बारे में मैं बताऊंगा कि 2012 में ये उदयपुर में यूनिवर्सिटी में वाइस चांसलर थे, तब मुझे इन्होंने चीफ गेस्ट बुलाया था कन्वोकेशन में, और वहाँ से मेरा एक ब्लॉक का उद्घाटन भी करवाया था। तो चहल साहब की जो तजर्बा है, जो योग्यता है, मुझे तब से मालूम है, तब से उनके संपर्क में था। और अभी हमारे केशव मौर्य जी ने सबके नाम लिए, मैं दोबारा नहीं लूँगा, पर मुझे सब पे बहुत गर्व है कि आप लोग आए। ये टॉपिक मैं पढ़ूंगा आपका एक बार फिर। एमिटी यूनिवर्स है, जो इसमें है इंटरनेशनल कॉन्फ्रेंस ऑन पैथोजन प्लांट हेल्थ एंड फूड सिक्योरिटी, रिसेंट एडवांसेस फॉर क्लाइमेट रेजिलियंस एग्रीकल्चर एंड लैंडस्केप कंजर्वेशन। ये एक ऐसा टॉपिक है जो हमारे भारत के लिए बहुत ही उपयुक्त है। इसको सुनते सुनते आज 10 विचार मेरे में आए इनोवेशन के, कि इस एरिया में, क्योंकि मैं तो आई एम मैट फॉर इनोवेशन। जिंदगी भर जर्मनी में ही मैं बहुत बड़े ग्रुप का रिसर्चर इनोवेशन का हेड था। तो मैं अपने विद्यार्थियों से, यहाँ बैठे हैं और भी यूनिवर्सिटी के लोगों से बात करूंगा। जब मैंने प्रोग्राम देखा शुरू हुआ तो मैंने पूरे यूट्यूब पर डलवा दिया। अब सारे हमारे वर्ल्ड के एल्यूमिनाई, 8 लाख एल्यूमिनाई सबको पहुंच गया और सब सुन रहे हैं। सारी यूनिवर्सिटी में, इंडिया में, जर्मनी में 28 स्कूल हैं, बाहर भी है, 20 कैंपसेस हैं। तो क्योंकि मैंने देखा कि ये तो एक ऐसा सब्जेक्ट है जिसमें हम भारतीयों को दिखाना है कि पूरे विश्व में इसमें हमारा कोई मुकाबला नहीं कर सकता। अभी जो माननीय हमारे मौर्य जी ने कहा कि जो भारत में बहुत बड़ा स्कोप है कि हम जल्दी 3rd रैंक में बनने वाले हैं, इसमें कोई डाउट नहीं है। अंडर द गाइडेंस एंड लीडरशिप अवर यशस्वी प्रधानमंत्री मोदी जी और यूपी के हमारे योगी जी के नेतृत्व में, अभी वो सिंगापुर गए और जापान गए, कितने लोगों ने वहाँ निवेश करा भारत में। तो मैं अपने स्टूडेंट आप लोगों से कहना चाहूँगा कि आप बिल्कुल ठीक रास्ते पे पहुँचे हो। 2005 में ये मैंने यूनिवर्सिटी बनवाई थी यूपी में। कुछ लोग कहते हैं कि सर आपको कैसे मालूम था कि उस वक्त ये यूपी नंबर 1 स्टेट बनने वाला है। मैंने कहा यही तो भगवान मुझे रास्ता दिखाता है, माय डियर स्टूडेंट। गॉड गिव्स अस द मोटिवेशन, शोज द पाथ, एंड वी आर गोइंग टू बी यूपी एज नंबर 1 स्टेट। अभी जब मैं देखता हूँ योगी जी, मौर्य जी और जो भी अधिकारीगण हैं, दिन रात लगे हुए हैं हर क्षेत्र में, प्रोडक्शन में, मैन्युफैक्चरिंग में, यूपी को नंबर 1 बनाना। और जो भी मेरे विद्यार्थी और शोधकर्ता, फैकल्टी मेंबर्स, लीडर्स लखनऊ कैंपस में हैं, सबका बहुत बड़ा सौभाग्य है कि आपको मौका मिलेगा ये दिखाने के लिए कि उत्तर प्रदेश पूरे विश्व में मशहूर हो रहा है और होता रहेगा। और आज का जो हमने प्रभावशाली और वास्तविक लेक्चर सुना, उससे सभी बहुत प्रभावित हुए। तो अब ये मैं अपने लोगों से बाद में वार्तालाप करूंगा कि इसका वो करवाएंगे हम लोग कि क्या लेक्चर से समझा, क्या सुना। अभी मेरे एसएनटी वार रूम मैंने बनाया है जिसमें कि 50 के करीब लोग हैं। उनका सिर्फ एक काम है तो ये देखना कि उसका आउटकम क्या होगा, क्योंकि हम लोग बहुत बड़े बड़े प्रोग्राम करते हैं। अब जो मेरे वॉरियर देख रहे हैं उनका काम ये है कि अभी इसके आप लोगों से संपर्क करेंगे, जो यूनिवर्सिटी वाले हैं, पूछेंगे कि इसको आप कैसे आउटकम में आपको हेल्प करें। पूरे विश्व में इसमें बहुत विचार आ सकता है, विचार बन सकते हैं। और जैसे हमारे ऊपर उन्होंने कहा केशव प्रसाद जी ने कि इतनी स्पर्धा है हमारे स्टूडेंट्स में, इतनी स्पर्धा है हमारे वैज्ञानिकों में। और मैं 28 साल जर्मनी रहा तो मैं इस बात को जानता हूँ कि जो हमारे भारतीय स्टूडेंट में, विद्यार्थियों में, पॉलिटिशियन में स्पर्धा है, वो मैंने विश्व में कहीं नहीं देखी। इसलिए मैं तो यही कहता हूँ कि हमारा हमारा देश पूरे विश्व का मार्गदर्शन करेगा। और आज कोई जो सब्जेक्ट है उनका भी मुकाबला नहीं है। और मैं ये जो हमारी शालिनी जी प्रोग्राम करती हैं वो हमेशा ही बहुत उत्तम रूप में करती हैं, पर मैं आज बहुत आश्चर्यचकित हूँ कि इन्होंने इस एरिया के सारे दिग्गज नेताओं को बुला लिया, दिग्गज साइंटिस्ट को बुला लिया। बहुत मैं उससे प्रोत्साहित हुआ हूँ। और अब मैं इनके मौर्य जी के बारे में बताऊं, जब भी मैं इनको किसी पार्टी में मिलता हूँ, किसी फंक्शन में मिलता हूँ, तो एक अपनापन नजर आता है इनका, इनकी योग्यता के बाद, इनकी पॉलिटिकल एक्यूमेंट के बाद। पर जो भी इनसे मिलता है वो जाके तारीफ करता है कि देखिए उन्होंने कितना हमें प्यार दिया, पहली बार में मिले हों। तो आज का जो ये प्रोग्राम है, इसमें मैं कहता हूँ कि बहुत हम लोगों को सोचना होगा, हम लोगों को नए नए आइडियाज निकालने होंगे। और जो आपने अभी नाम लिए केशव प्रसाद जी ने, उन सबको मैं अब दोबारा नाम नहीं लूंगा, पर मैं बाद में आप लोगों से बात करूंगा, मिलूंगा, क्योंकि ये आप लोग एक मार्गदर्शक हो इसके लिए। और अब मैं एक दो बातें बताना चाहूँगा आपको एमिटी यूनिवर्सिटी के बारे में। अब हमारा 12 कैंपसेस हैं कंट्री में, साउथ में हैं, सब तरफ हैं, और मेरा एम है कि हर स्टेट में एक कैंपस हो। और विश्व में जो हमारे अभी 18-20 कैंपसेस हैं, तो मैं 100 कैंपस बनाने जा रहा हूँ, क्योंकि जो भारत का विचार, जो भारत का जो विचार और जो आइडिया है, 28 साल के बाद मैंने देखा मैं क्यों इतना बड़ा बन गया वहां पे, साइंस एंड टेक्नोलॉजी कॉन्ग्लोमरेट का। और जब मैं बताऊं आपको स्टूडेंट्स, माय स्कॉलर, ईस्ट और वेस्ट का जब यूनिफिकेशन हुआ 89 में, मैंने अपने लोगों से कहा कि मैं इसमें सबसे बड़ा इन्वेस्टर बनूंगा, सबसे बड़ा टेक ओवर किंग बनूंगा। तो सब लोग हंसे तो नहीं, क्योंकि बॉस के सामने हंसना तो नहीं हो सकता ना, पर मैं अपनी तारीफ के लिए नहीं कह रहा, मैं एक भारतीय की तारीफ के लिए कह रहा हूँ, अपने स्टूडेंट की तारीफ के लिए। आई बिकेम द बिगेस्ट टेक ओवर किंग। चांसलर कोल ने कहा कि मैं तो पॉलिटिकल यूनिटी लाया हूँ, पर चौहान इकोनॉमिक यूनिटी लाया है। मैं इसलिए कह रहा हूँ स्टूडेंट, आप एक बार जैसे हमारे आदरणीय मौर्य जी ने कहा, निश्चय कर लो कि क्या करना है, उसके पसीनों पर चलो इस निश्चय के, और कोई आपको रोक नहीं सकता। हमारे जो भी इस वक्त 2.5 लाख स्टूडेंट पढ़े हैं हमारे यहाँ, 2.5 लाख स्टूडेंट पढ़े हैं। ये सब हमारे प्रेसिडेंट के, फाउंडेशन के प्रेसिडेंट अतुल चौहान जी और वाइस प्रेसिडेंट असीम चौहान जी का योगदान है। ये सब लोग बहुत बड़े बड़े पदों पर यूरोप में थे, परंतु जब उन्होंने देखा कि पापा का एक उद्देश्य है भारत को खाने का, तो सब कुछ छोड़ के यहाँ आ गए। तो मैं ये कहना चाहूंगा कि जैसे इस वक्त हमारे पास जो NIRF का रैंक होता है, उसमें हम 22 नंबर पे हैं 1200 यूनिवर्सिटीज में। तो यह दिखाता है कि जो हमारे अभी माननीय केशव प्रसाद जी ने कहा, उस पर हम चरितार्थ करेंगे, उनसे प्रोत्साहित होकर। जो हमारा QS रैंकिंग है, वो हमारे हम लोग अमंग द टॉप यूनिवर्सिटीज हैं, लाइक IIT एंड ऑल, IIT मुंबई। तो मैं इसलिए कह रहा हूँ कि माय डियर स्टूडेंट, रिसर्च स्कॉलर्स और माननीय जो हमारे गेस्ट आए हैं, उनसे रिक्वेस्ट होगी कि हमें आगे मार्गदर्शन करें, आगे हमारा रास्ता दिखाएं। हमने 55,000 हम लोगों ने अभी टोटल 55,000 हमने पेटेंट फाइल कर हुए हैं, पब्लिकेशन है 55,000, और पेटेंट भी हमारे बहुत बड़ी मात्रा में हुए हैं। और 35 टेक्नोलॉजीज वी हैव ऑलरेडी गिवन टू इन डिफरेंट एरियाज। तो इस तरह से मैं ये बताना चाह रहा हूँ कि अगर माय डियर स्टूडेंट एक निश्चय कर लो, जैसे हमारे माननीय मौर्य जी ने कहा, तो उस निश्चय को रोकने वाला कोई नहीं। अगर आपका संकल्प हो, तो मेरा एम है कि हम एमिटी यूनिवर्सिटीज को, हम एमिटी यूनिवर्स कहते हैं, पूरे विश्व में एमंग द टॉप यूनिवर्सिटीज लाऊं, उसी रास्ते पर चल रहे हैं। हमारे माननीय मंत्री वाइस हमारे मौर्य जी आए, तो ये इसलिए आए कि उन्हें पता है कि यहाँ पर जो मेरे विचार है वो चरितार्थ करेंगे, उसके लिए लोग संकल्प करेंगे। तो मैं ज़्यादा टाइम नहीं लूंगा, मैं यही कहूँगा कि मेरा करबद्ध सबसे प्रार्थना है कि हमें मार्गदर्शन करिए, हमें बताइए, और उसको चरितार्थ करने के लिए मैं हूँ, अतुल जी हैं, असीम जी हैं, पूरा परिवार लगा हुआ है। एक ही एम है, एक ही निश्चय है कि भारत को सुपर पावर बनाना है 2047 में। और मैं अपने स्टूडेंट के साथ डांस करूंगा तब, चाहे कोई भी एज हो। तो भगवान ने मुझे कह दिया 106 साल की तो मैं देता हूँ आपको, अगली बात उसके बाद करेंगे। मैं इसलिए बता रहा हूँ कि एज वेज कुछ नहीं करती, अगर निश्चय है तो भगवान को आपकी जरूरत है। ये हमारे माननीय मौर्य जी को, भगवान को उनको जरूरत है। जो जो आज बोले, जो प्रचार प्रकट करे, सबको जरूरत है। मैं सबका बहुत आभारी हूँ। परम आदरणीय केशव प्रसाद मौर्य जी, आपका मैं बहुत स्पेशल आभारी हूँ कि आप आए, टाइम निकाला। और हमारे जो वाइस चांसलर अनिल विशिष्ट जी हैं, और हमारे जो शालिनी सिंह हैं, ये तो कमाल का ही काम करती है, जो भी प्रोग्राम करती है। तो इनकी जितनी तारीफ को उतना कम है, पर मैं ज़्यादा टाइम नहीं लूँगा। सबके लिए मेरा हार्दिक आशीर्वाद। बहुत प्रोत्साहन मिला, बहुत प्रेरणा मिली, और उत्साह मिला कि जो मेरे विचार हैं उनको कार्यान्वित करूँ। और मौर्य जी, बहुत बहुत मेरा करबद्ध प्रार्थना कि हमें गाइड करिए, आशीर्वाद दीजिए। हम लोग दिखा देंगे आपको कि एमईटी यूनिवर्सिटी जो है उसका दुनिया में मुकाबला नहीं है। उनका दुनिया में कोई इतना कर नहीं सकता जो हम कर रहे हैं। असीम जी ने बोला तो ज़्यादा बोलने की जरूरत नहीं है। तो जब बेटे ही इतने काबिल है तो फिर बाप को सुनने की जरूरत भी नहीं होती। ये तो एक परंपरा है। अतुल जी, असीम जी और सारा परिवार वार्टन के हैं, हार्वर्ड के हैं। राइस यूनिवर्सिटी में अतुल जी की बेटी पढ़ने गई हुई है, तो वो कह रही थी वहाँ एक गार्डन है, एग्रीकल्चर गार्डन, उसमें मैं कोई नया प्लान डेवलप कर रही हूँ। तो मुझे ताज्जुब हुआ कि राइस में जो यूनिवर्सिटी का गार्डन है उसमें कहती मैं एक नया प्लान डेवलप कर रही हूँ। मैं इसलिए कहना चाह रहा हूँ कि आप हमें गाइड करो, हमें मार्गदर्शन दिखाओ, और हम आपको करके दिखाएंगे जो मैंने कहा है वो सब चरितार्थ होता है। केशव प्रसाद मौर्य जी, आपके लिए बहुत बहुत धन्यवाद देता हूँ। आप हमें आशीर्वाद देते रहिए, मार्गदर्शन देते रहिए, करता हम होंगे, करेंगे। धन्यवाद।
We need your thundering applause for our Honorable President, sir.
केशव प्रसाद जी करो, ठंडी में रहिए।
Thank you very much, sir, for your presidential address. Sir, this was the address. There is a felicitation, sir. If you could give 5 minutes, sir, more. हम निवेदन करेंगे। धन्यवाद माननीय उप मुख्यमंत्री जी, अपना समय थोड़ा सा और देने के लिए। मैं फाउंडर प्रेसिडेंट सर को बस एक ही बात कहना चाहूंगा। एक शेर है, मैं अकेला ही चला था जानिबे मंजिल मगर, मैं अकेला ही चला था जानिबे मंजिल मगर, लोग साथ आते गए और कारवां बनता गया। एमिटी का ये कारवां आपके नेतृत्व में 2047 में आपके कदम से कदम मिलाकर विकसित भारत की ओर बढ़ेगा। हम आपको पुनः ये आश्वस्त करना चाहते हैं और करतल ध्वनि से पुनः आपकी तो ध्वनि पहुंचे कि एक विकसित भारत के निर्माण में हम सभी पूरी दृढ़ता के साथ आपके साथ खड़े हैं। नाउ फॉर very quick felicitation award, I would request Professor Pokhar Chahal, Secretary ISMPP, to come, and we have Honorable Deputy CM sir has very limited time. Yes, please. Thank you.
बहुत बहुत धन्यवाद सर। सर, आज के कार्यक्रम के मुख्य अतिथि आदरणीय केशव प्रसाद मौर्य साहब का मैं इंडियन सोसाइटी माइकोलॉजी प्लांट पैथोलॉजी की तरफ से हार्दिक अभिनंदन और स्वागत करता हूं। सर, आप एमईटी यूनिवर्सिटी में आईएसएमपीपी की इंटरनेशनल कॉन्फ्रेंस में पधारे, आपका बहुत बहुत आभार और बहुत बहुत धन्यवाद सर। हम इंडियन सोसाइटी माइकोलॉजी प्लांट पैथोलॉजी की तरफ से सर देश के जाने माने वैज्ञानिकों को सम्मानित करते हैं। इसी क्रम में आज हम हमारे ऑनरेरी फेलोज को सम्मानित करने जा रहे हैं। इसके लिए मैं हमारे डॉक्टर AN मुखोपाध्याय साहब, डॉक्टर CD मई साहब, डॉक्टर C मनोरिया साहब और डॉक्टर SS चहल साहब को अपने स्टेज पे आमंत्रित करता हूं कि आप सर पधारें। और हम पहले ऑनर दी
देश के शीर्षस्थ वैज्ञानिकों का सम्मान हो रहा है, कृपया करतल ध्वनि से स्वागत करें।
सर इसी क्रम में मैं अब सोसाइटी की तरफ से वायलिन आउटस्टैंडिंग प्लांट पैथोलॉजी टीचर अवार्ड 2025 के लिए डॉक्टर कृष्णा प्रताप सिंह, प्रोफेसर एंड हेड डिपार्टमेंट ऑफ प्लांट पैथोलॉजी, GBPOT पंतनगर से निवेदन करूंगा कि आप स्टेज पर पधारें और ये अवार्ड ग्रहण करें। और दूसरा अवार्ड प्रोफेसर SS चहल लाइफ टाइम अचीवमेंट अवार्ड के लिए मैं डॉक्टर अश्विन कुमार बसंत राय सर, जो फॉर्मर डीन कॉलेज ऑफ एग्रीकल्चर एंड कॉलेज ऑफ बेसिक साइंस पालमपुर से निवेदन करूंगा कि आप भी पधारें और प्रोफेसर SS चहल लाइफ टाइम अचीवमेंट अवार्ड ग्रहण करें।
This is Lifetime Achievement Award. Please give a big round of applause to sir. May I request the dignitaries to take the seats on the dais? We are just concluding. After the photograph, I can begin. I'd request Dr. Jameel Ahmed, Principal Scientist, Division of Plant Quarantine, to give his words of gratitude. Sir.
Thank you so much. This is ultimately the almost end of the inaugural session. Honorable Deputy Chief Minister Shri Keshav Prasad Maurya ji and distinguished guest of honors like Dr. C.D. Mai Sahab, Dr. S.S. Chahal Sahab, Dr. D.R. Singh, Vice Chancellor, Bihar Agriculture University, Professor Anil Vasisht, Pro Vice Chancellor, Lucknow campus. Then we have the organizing secretary, Professor Dr. Salini Singh Bisain, and we have eminent scientists from all over India and at least 12 expert speakers from different countries, and chairman this this university and the founder president of this university, Shri Ashok Chauhan ji. I wholeheartedly thank for giving us the opportunity to organize particularly the international conference. This is the first time we are organizing this particular conference in any private organization. This is the first time. So I thank everyone for giving your time, and sir, Deputy CM sir, for your information. We have the legendary plant pathologists from the country like N. Mukhopadhyay sir, Dr. C. Manorachari sir, then we have Dr. G.B. Singh sir, so many. I don't want to take the name. Then ultimately the entire team of this particular campus in organizing this particular session in a very defined manner. So I heartily thank everyone without taking the name, because of paucity of time, we have to just end the session. And if I forget the name, Please pardon me from the authorities from this particular institute, then the persons from media and press. I once again thank everyone. Thank you so much.
सर, हम राष्ट्रगान के लिए खड़े होंगे, बस राष्ट्रगान के लिए मैं आप सभी से अनुरोध करता हूं कि कृपया राष्ट्रगान के लिए खड़े हों। कार्यक्रम का समापन राष्ट्रगान द्वारा।
जय हे भारत भाग्य विधाता, पंजाब सिंध विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा, तव शुभ नामे जागे तव शुभ आशीष मांगे, गाहे तव जय गाथा, जन गण मंगल जय हे भारत भाग्य विधाता।
भारत माता की। थैंक यू वेरी मच। इसके बाद आप सभी लंच के लिए आमंत्रित हैं और बाहर ग्रुप फोटोग्राफ्स के लिए भी सम्मानित वैज्ञानिकों से और आपसे निवेदन है कि ग्रुप फोटोग्राफ के लिए कृपया बाहर आ जाएं।
सबको ये होगा सर को देगा जब सर का प्रेजेंटेशन, राइट? आई नीड अ माइक। सर आप फिलहाल ये सर ही प्रिपेयर करें, बाद में हम चर्चा करें। जी सर। चल रहा है ना?
हेलो।
Is it all right? Good afternoon, everybody. We are going to have a very, very important session. It's a plenary session, and I could see there are very renowned speakers available for this presentation, but I am a little upset that the attendance is very poor. When you are in a plenary session, you know, they are all delivered by very eminent and experienced people of the country and abroad. But anyway, we'll start with Dr. Garrett, Caria A. Garrett. He's the preeminent professor, University of Florida, and he was the immediate past president of American Phytopathological Society, very renowned. She's the preeminent professor, University of Florida currently, and immediate past president of American Phytopathological Society. She'll be online, and the topic of his lecture, our lecture, is scaling epidemiological models in new plant health program management ecosystem. Scaling epidemiological models in new plant health management ecosystem. She's on the line. So can I request Dr. Garrett to present her lecture?
Yes, thank you. How is the sound? Can you hear me okay?
You are okay, Karen?
Okay, yes, thank you. Just a moment, I'll share my screen. Let's see, does that show up well for you? Does the screen show up okay for you?
Online is okay.
Okay, great. Thank you. Yeah, and thank you so much for the opportunity to present at this meeting. It's a great honor, and I appreciate it. I wish I could be there in person. But so today I'll be talking about scaling epidemiological models. So thinking about how to scale up epidemiological models and beyond, how to think about larger scale impacts from projects. So a thought exercise for you. As I'm discussing this today, so you could think about if you were developing your project so that it supports the implementation of a linked comparable project in every country. So that's one thing that we've been thinking about in terms of how we can develop projects for a particular context, but then make them more general and through developing software and models that we can use across different systems. So that's one aspect that I'll be talking about today. One of the inspirations for me in thinking about plant health is this concept of a global surveillance system for crop diseases. So the idea here that there'd be a network of countries that would be working together for surveillance and beyond surveillance for mitigation as well. And of course, for all countries to be a good partner in a system like that, there are a lot of different sorts of capacity that we could build globally, risk assessment networks, communication networks, and so on. So that's one inspiration for what I'll be discussing. So this paper has some views on a model of how to implement a system like this from an epidemiological standpoint, especially. So in this sort of ideal system that we'd like to have in place, we would have data layers such as for crop losses, global crop loss estimates, weather climate maps, microbiome distributions, cropland density, et cetera. Of course, we don't have good data layers like that for all the types of data that we would like, but so thinking about what would be a good system to have in place. And then we'd like to have models, for example, models of disease response to weather, image analysis, image libraries for predictive models, models of loss due to disease, et cetera. And then to have those translated into decision support tools that can be used for example, surveillance and mitigation prioritization. And then for those to be linked into a global system of the stakeholders important for crop health. And again, accumulating and then distributing data again to keep strengthening these data layers, models, and translation of the models. So of course, we don't have all that in place right now. So one thing that I'll be talking about is some steps to getting a sort of national plant health risk analysis for each country that can link into this sort of system. Another perspective is thinking about disaster plant pathology. So here we're looking at how to respond to different types of disasters for protecting plant health. So for example, pulse disasters like flooding or droughts. Floods, as we know, can spread propagules of some pathogens. And so if people are prepared, they can have a better response to the risk from that. And then pest disasters like climate change or different types of civil unrest can be slower moving in some cases, but then also systems need to be able to adapt to them. For example, to make sure that seed systems can prevent the spread of local pathogens and especially introduction of new pathogens. And then I'd like to mention this project in the context of climate change. I think it's an unusually interesting project in some ways because it is it's incorporating 27 crop pathogens and pests in a simultaneous analysis for comparison across 4 crops. And in this project, Romaric Mafo-Chinda and colleagues use machine learning to identify environmental predictors for each of these crops, and including some interesting types of new predictors like cropland connectivity, as well as crop host density. And I think one thing that's interesting about this is that based on climate matching, if we're looking across lower altitudes and higher altitudes, we find an estimate here of 44% of the pathogens and pests that could become more common with warmer temperatures, but then 17% that may become less common. So I think that's an important point as well, that at the same time that in this case, we're estimating that around half of the pathogens and pests could become more common, but then some share will become less common. So I think that's an important point for growers as well, to be able to anticipate which of the profile of pathogens and pests will become a more important problem in the future. So now the toolbox that we've been developing, as I was saying, to scale up, to think about how we could learn from past projects to improve future projects. So this R2M Plant Health Toolbox, it's designed to make the most of existing data and knowledge for rapid assessment. So if we're looking at how to support mitigation of crop diseases and pests. So, and this is a QR code to access some information about this toolbox. So it includes a set of tools. So for example, if there are missing epidemiological parameter estimates, how to improve expert knowledge elicitation. We have an app to make it simpler to implement expert knowledge elicitation, how to improve that to get good parameter estimates. And then a habitat connectivity analysis to help answer the question of what are key locations in the analysis of potential invasion networks or epidemic networks. An app in progress for trade analysis to look for priorities for monitoring trade. And then a scenario analysis to answer questions about the best regional strategies and tactics. So I'll tell you a little bit more about those tools and how they can scale up. And so again, the idea that if any given analysis that these tools are used in, we can learn from and keep improving them for future analyses to get a positive feedback for future studies. So expert knowledge elicitation. So this is an article describing some of the pros and cons of expert knowledge elicitation by Jacob Robledo and colleagues that was in a phytopathology special issue recently. So it discusses, I think, the interesting idea about how expert knowledge that is from sources that are not digitized fits in with the universe of knowledge, right? With artificial intelligence linked with expert knowledge. So of course, artificial intelligence is accessing digitized materials, while experts are aware of a lot of unpublished information, their own unpublished observations, and their discussions with other experts. So expert knowledge elicitation is a way to help integrate expert knowledge into the system and also make it available for summaries from artificial intelligence. If you're familiar with Bayesian statistics, this is one way of conceptualizing how expert knowledge fits in. It could be the prior knowledge about a parameter like infection rates. And then when new experiments can be implemented, those can build on the knowledge to summarize it in a posterior. So in the context of Jacobo Robledo's work with Fusarium wilt risk due to tropical race 4, he implemented a really extensive expert knowledge elicitation with lots of different groups of regional experts in Colombia. And for those of you who work with Fusarium wilt, I'd be happy to talk to you more about this study. Another tool is for analysis of cropland connectivity, or more broadly, habitat connectivity. So if you're starting to study the potential invasion of a region, this can be a first step to get a baseline understanding. Where habitat is available, there's more likely to be an emerging pathogen or pest. And where habitat is connected to other habitat, there's even more likely that that region will have an important role in epidemics. So to make it easier to look at habitat connectivity, Aaron Plexula and colleagues have developed an R package for mapping habitat connectivity. So the idea here to make it easy using existing data sets to look at habitat connectivity, especially there are a lot of existing data sets for crop-specific pathogens for the crops that are important for them, for example. And then the habitat could be expanded to include climate-based habitat information. Then another perspective on scenario analysis to understand management strategies. So for that, we've worked on an R package for impact network analysis. So that's available now as an R package, and working on an online app for non-R users as well. So for example, if you have networks like this, suppose the nodes in these networks are farms or political units at a larger scale, and then the links indicate the potential for movement between them. So in the right-hand side, you can see that which nodes are going to be particularly important in this. Sometimes it's more subtle, as in the other cases. And of course, if it's a really big network, then an analysis is necessary, since just considering it visually isn't sufficient to understand the roles of the different locations in the network. And then in impact network analysis, we're combining like the lower, in this multi-layer network, the lower layer is an epidemic network or an invasion network. And then we're also considering with that a socioeconomic network, where each of those nodes or locations represents a manager making a decision, in this case about adoption of a management approach. And so those managers then are linked in to the epidemic network, where if managers decide to use effective management, they're slowing the spread in the epidemic network. Or if they don't manage, then they're making it easier for epidemic spread to occur in the epidemic network. So then the combination of these different networks determines what's happening in a region. So you could use a scenario analysis like that to ask questions like, what locations are particularly important? How are the benefits of the system distributed? And then also to consider different potential subsidies and policies and how they would affect the system outcomes. If you'd like to see an example of this kind of network, here's an analysis for avocado laurel wilt. So in Florida, this was a new epidemic in the past decade, first spreading in wild plant populations and then in avocado populations. And so it's interestingly complicated story, but one of the take-home messages from this analysis is about the potential role of hesitancy to manage at the beginning of an invasion by a pathogen. If a lot of growers are thinking that maybe they can wait to manage or be cautious about the pathogen, then that will speed up the epidemic, right? So that's where policies can be particularly impactful if they give growers incentives to be active early in the epidemic to slow down the spread so that it's not such a rapid epidemic. So using these tools, we're thinking about, as I mentioned, a national plant health risk analysis, how to define that. And again, thinking in the global context, how could we set that up so that every country could be implementing a national plant health risk analysis for their key crops. So here's an example for Cameroon and Ethiopia of a start toward conceptualizing this national plant health risk analysis and how to implement it in a rapid way and being as effective as possible. Like I said, we keep thinking about how we can improve this as we develop new applications. And here's a more specific example using some of the same data in Ethiopia for managing bacterial wilt, some emphasis on that, and then also more generally potato pathogens. So this is an illustration, if you would like to check that out, of some of the ways of integrating these different analyses. So to conclude, I'll just mention, as we're thinking about this national crop health risk analysis and how we can implement it across countries and ultimately having a set of tools that any country could use and as part of a network, a global network for surveillance and mitigation. So we would welcome the opportunity to explore national crop health risk analyses for other countries. Well, for example, India and other countries that you work in. So here's my email address if you would like to follow up with questions about that. I'm happy to answer any questions now if there's time, or afterwards through email or other means. Yeah, and so thanks again for the opportunity to present, and I hope you have an excellent meeting. Thanks.
Thank you, Dr. Garrett, for your excellent presentation. It's an eye-opener for many of us that how she could coordinate and got involved so many scientists at a time for such a beautiful analysis. I think this is how we should work out. I could see in some of the publication, there are more than 25 or 30 authors. So they are all contributing in their own way, you know. So if you want to manage such a complex problem, there is no other way out but to get involved. Many people, not only from your country, but across the world, so that you could get the correct data and identify. I'm very grateful for this wonderful analysis, and I'm sure from India, somebody should be interested and get in touch with you for any further collaboration. Any question or comment from the audience, please?
Can I?
Yes, please.
First of all, Dr. Karan, it's a wonderful presentation as the chair has been beautifully narrated. I have that privilege to work or collaborate with you in a distinct way. But your cropland analysis and also understanding the various epidemiology is going to be the key driver for the next several years to come, especially when we see that viral disease, fungal disease, bacterial disease, each has its own dynamics in view of climate change. Perhaps national systems and private companies and several people have to come together and especially join this kind of bigger network. I'm sure you have a bigger vision for the global systems, and we are very hopeful that your system we are going to get benefited. And we are very thankful and very honored to have your lecture today. And it is wonderful for all of us to have you here. Thank you for nice presentation, but I will be in touch with you closely as used to be. Thank you so much.
Thank you.
Thank you. Any other interaction? Any other comment? So I think India being such a vast country and varieties of crops grown all over the country, especially the problem which is coming up for RES4 in banana, I think a lot of things can be done in that regard, otherwise we may face a serious problem where there is no solution to that. So possibly it is time for us to interact, cooperate, and coordinate with others rather than working in an isolated compartment. I know banana RES4, that is, which is a very serious problem, as Dr. Mai was mentioning in the morning. So let us talk about some other problems. And I'm told that India has now taken up a pulse mission, spending huge money because we are importing a lot of pulses from other country. But they have mentioned about a pulse mission, but I don't see that they're going in the right direction, involving people and taking their opinion about it. If you are working on the pulse, you should take the people who have worked in pulse for last 20, 30 years, take their opinion, take their advice and go. But I don't know, we are working in isolation. Possibly there is a need to get together the people, older people, younger people and others, so that we can make this pulse mission very successful. Like that, such type of work which is presented by Pam this morning is possibly an eye-opener. And I think we should collaborate in a more extensive way, joining hands with other people so that we can come out with a solution. So with this, I thank you very much again for your wonderful presentation. And thank you. And I'm sure the people will be benefited. Next speaker is Dr. Chahal. Is he ready? Dr. Chahal is here. Dr. Chahal is a good friend of mine for last 3 or 4 decades. I have the pleasure of watching him, performing him at various occasions. He was a great student at PAU, then he was registrar, then he was vice chancellor at Udaipur, and he was president of the Indian Society of Mycology and Plant Pathology. And in fact, he's a father figure, and he always tried to see the ISMPP is in right direction. So his experience is not only for science, but his fellow feeling for others is very important. And his topic of his talk is very interesting. He said, what led us in last and what will lead us in the next 25 years in plant pathology? This is a big question. At one time, we were talking about plant pathology. In the process, we forgot many things that I point out every forum, every occasion, that we are forgetting mycology. Dr. Monarachari is here. He's the doyen of mycology in the country, but there is no importance in mycology. There is no importance in taxonomy. Because how can you talk about plant pathology unless you know about the pathogen, know about the species which is creating the problem. So it has totally died off from the plant protection area. So like that, what Dr. Chahal is saying, that what is there now and what is to be done for the future. So I'm very particular to talk about it. I've talked in various occasions that we should give importance to mycology, or taxonomy, because without the basic knowledge of that, nothing can happen. But you'll be surprised to see that in ASRB or in case of agriculture science sector, there is no position of mycologist. So the students are not going for mycology at that way. So Dr. Chahal, I request you to give your bright ideas for the present day pathology and the future of plant pathology. Thank you.
Thank you, Mr. Chairman. Most respected Dr. Mukhopadhyay ji, I think mentor for many of us, and my dear friend, Dr. Basandrai, congratulations to you for achieving this award. And what can be beautiful moment for me, whose student has achieved a Lifetime Achievement Award, and he is chairing my session as well. Dear participants, I am privileged to deliver this lecture in this conference, which is in fact focusing on the recent issues in advancements in agriculture and management of plant pathogens. I thought it worthwhile to consider this and identify directions that we followed in the last 25 years and what we expect that will lead us in the next 25 years in plant pathology. Mainly the purpose of this, that many youngsters, how they should prepare their project and to which directions our plant pathology is being taken up. Now, to make a fair assessment to how this single cut and burn technology that turned into an interdisciplinary cutting edge science, that science-based discipline more to say, we brought out one treatise in some 20 years back. And this was published in 2006. And we reviewed that the progress in 100 years in that volume. What we found that was from very simple identification of plant pathogens, there was sound base of diagnosis that was developed in this duration, and understanding of complexities of host pathogen interaction, ecology of ecology, recurrence. Then several laboratories, they have come out, state of the laboratories came up in the country. There was advanced research that was carried out in this century that was on frontline area. And there was, of course, unfolded and astonished behavior of pest pathogen that was understood. And this was, of course, there was devising novel control measure for these different plant diseases. This happened because of this, that in 1905 in India, this agriculture education that was established. And in the previous century, we could achieve a lot of this in the direction of plant disease control. As Mr. Chairman has explained in mycology, it's an important component of plant pathology, what we consider. It is rightly, he has said that it's a little bit neglected in these years. But in last 25 years, mycological research, that have been, of course, there was profound shift from moving from foundation in mycology, and it was more towards the taxonomy. It was, they were taken up. But it has moved towards a high throughput molecular base and molecular omics discipline that has been taken up. There was widespread adoption of DNA sequencing in the last 2 decades. Fungal classification that has been, of course, redefined, and DNA sequencing that led to major reclassification of fungal groups. Generally, we say this as a polygenic overhaul in this fungal biology. Fungal genome, that mapping has been taken up, and this is widely, it has been expanded, and more exponential, this expansion that has been seen in exploration of fungal biodiversity. Now, it has changed the understanding of fungal role in global adaptation. And what we see that in next 25 years, next 2 decades, how we are moving towards this, there is likely to be shift from traditional taxonomy towards high-tech application of biotechnology. And this is from disciplines, these descriptive studies to more to critical, and it will become a mega sciences that we are expecting. and fungal adaptation and survival of these pathogens under global warming that will draw our attention in the coming years. Then researching fungal pathogens and which are threatening our food security. That is, of course, coming up in a big way. And exploring unknown species biodiversity, of course, this has already been taken up. This is likely to be continued in the next 25 years. What we are seeing in plant diagnostics, research in plant disease diagnostics has undergone a transformative shift from traditional laboratory-based assay to rapid and non-invasive and digital technologies. What we have seen that in key advancement, there has been taken up in molecular acceleration, then pre-symptomatic studies, these have been now given emphasis in this LOP, this VOC walk, this has been now taken up in different laboratories. Currently, there is more use of this artificial intelligence tools, more use of robotics, smart sensing, and then the CNN, and then, of course, image analysis and UAV, unmanned aerial vehicles, and smartphone. There its use that has been now taken up in a different way in different places. What we see in application that has this AI application that has transformed disease detection and diagnosis, this system that is catalyzing innovation in many ways, and better disease management that has been expected and being taken up. Many studies, these have been taken up, these have been carried out in application of AI in plant pathology. Of course, it offers disease detection and diagnostics, which is rapid and precise now. For example, this CNN technology, with this, in previous lecture, you have seen that cassava diseases, these have been taken up. Now, in cassava, there are so many diseases, and it's a disease complex. Identification is very difficult. But with this technology, 90% accuracy that has been achieved. Similarly, diagnosis of wheat blast pathogen with machine learning that has been taken up in a big way, and accuracy is 95% in diagnostic. So there are many other examples also. I have taken just few. But as you see in the tomato, for example, it suffers from number of serious diseases, tomato, borers, caterpillar, whitefly, and many others, of course. It is difficult, it becomes complex. So this in this case, this deep learning based approach that has been taken up, and camera devices that has been applied for with various resolution to identify these diseases with high accuracy. It is offering, of course, it's being used as immense useful, it has been found. The similarly detection of this virus, grapevine, that is difficult in many ways, but it has been taken up and it has identified early stages. Potato leaf blight. This is a very serious disease, a worldwide occurrence, and it has potential to cause swift and complete loss. In this case also, this has been applied, and successfully this early detection of diseases that have been taken up with this technology. Now, studies have been made in the classification of paddy leaf diseases using deep learning algorithms, and various models these have been developed. There are so many models, but out of this, efficient net BO that has been seen as the best with 97% accuracy. This has been applied in paddy also, and then CNN that has been applied in wheat rust, and similarly in case of other diseases, it has been taken up. And what we have seen that in plant disease diagnostic in next 25 years, what we expect. What we expect in this case is that it's swift, of course, from laboratory to real time. That is a challenging practice, and it will be seen. But it is expected that it will be a point of care solution. There will be device with these technology. There will be advanced CRISPR-enabled molecular diagnosis, and there will be hybrid AI deep learning advancements in beyond CNN. That will be taken up, and there will be smart data set optimization that is desired. But what is expected more in this case is that at this stage, this AI is not so much interpretable. What we are expecting now is XAI, that is offering, of course, interpretability more and more. So in this way, this multi-model sensor fusion, that is very important. And these biosensors plus nanotechnology for rapid disease detection, that will be, of course, taken up in this. In this, I must say, the sensor-based technologies, they are now being implemented for monitoring pests and pathogens more and more, which are difficult in the many crops, but now it is correct and accurate, and it's more practiced. So this may these be weather sensors network, image acquisition, natural pests, or other, but new this precision based system that's being now developed. I hope Professor Mai, Dr. Mai, he will be taken up this very aspect in a very different way, because this is in cotton. We have seen that it's in detail, these algorithms, these have been applied for sucking pests. So this, of course, is a very, but more emphasis is likely to be on pre-symptomatic diagnosis and use of course, microbiomes, that is this. But what we expected that this global pest disease diagnostics market that is projected to be grow significantly in the coming years. Then come to host resistance. During last 5 years, host resistance that has been research transitioned from relying just solely on classical plant breeding. That is implying just advanced genome tools. And in this deeper understanding to plant pathogen interaction that has been taken up, of course. But key achievement is when we say in resistance, host resistance, that we can just sum up, this is a hybridization, MAS, that has been applied for identification of R genes. Adaptation of new generation sequences, NGS, what we call, that has been taken up. CRISPR is taken in a large way, gene editing. Then broad spectrum resistance that has been also taken up, and disruptive S genes that has been taken up, and RNA interference. This is being practiced for durable resistance, emphasized more and more. So this we have directed towards climate change resistance that has also been taken up, and bioinformatics, this AI to predict gene function, that is all. But what we are to which direction this resistance mechanism that is likely to go is that there will be more emphasis on this designing synthetic immunity, more and more. Many laboratories, they are already focusing on this. It is expected to shift towards durable broad spectrum AI strategies that will be developed, combining host resistance with the ecological practices, because with this climate change, that is a big challenge. Developing non-host resistance, that is actually likely to be practiced more. In chemical control, there is important aspect. There was sometime this was very important aspect in plant pathology, chemical control. We can say that there was era of chemical control also. This plant pathogen last 2 decades, these have been, of course, this control has evolved, relying more and more on broad spectrum for these products, utilizing sophisticated and targeted compounds. Amongst these is last synthetic fungicide, broad spectrum fungicides, then increasing pathogen resistance that has also come up to manage this. This was focused. Use of one shot. This technology was developed first in Japan. This was also practiced in the last 2 decades. There was significant growth of research and development and investment in major companies that was attracted in this era, in this industry. More emphasis was on plant-based fungicides, strobilurins, then triazoles. It remained dominant. Dominant, of course, botanical pesticide. These have been taken up. Nanoparticles, these have efficiency that has been taken up. What we expect, how it is likely to go further is that there will be more emphasis on developing high selective and safe synthetic molecules. Further, it is expected that this balancing of that will continue the necessity of synthetic fungicide with the urgent need of sustainability and this more sustainability of resistance mechanism and climate change adaptation that will draw our attention. And of course, SDHI, triazole, it will continue, and this more emphasis on systemicity, how it can be enhanced. Systemic fungicide, they are right, but how this systemicity in plant that can be enhanced, that is likely to draw the attention. Biocontrol chemical integration, that is important now being taken up. It is being considered that when this, it can be very useful. There is likely to be hybrid fungicides combining botanical extracts and these conventional chemicals. So this, coming to this biological control, the beginning of this century, biological control, that was a small scientific interest. But during last 20 years, it has become an important component of sustainable agriculture. There was shift solely from that was relying on single antagonist to developing this microbial consortia. Use of identify different species, mycoviruses, and this Bacillus, Pseudomonas, Trichoderma, these are, of course, main species which are used. Walk, this was definitely a part, and then use of omics, then nano and biopriming techniques. These were developed. And there was robust formulation of granules and smart treatment. So it is practiced in this way that what we can expect is this likely to become a mainstream part of advanced IPM. There is likely to be AI-driven microbial selection of BCA, CRISPR technology that is will be empowered. And this expanded use of microbiome that is now expected to be taken in a big way. Then integrated disease management, how this is moving in plant disease control, that IDM that is has evolved from traditional chemical heavy approach and data driven to holistic strategy. In last 25 years, what we expect is there was a usually spray to calendar module that has been employed, emphasis on host plant resistance, expanded biocontrol, and global policy integration that was taken, but more focus on disease forecasting, nanofungicides, cultural and judicial use of chemical, these were emphasized. Where we are moving in IPM, the next 25 years will likely to see IDM and likely to get higher this momentum on eco-friendly approaches. This is likely to reduce 20 to 30% chemical in a big way. It is expected to shift in that way. There will be rapid growth of biopesticide, biointensive IPM, and there will be more interdisciplinary approach in this era than when climate smart IPM, use of molecular tools, these will taken up, and IPM market, particularly in next 2 decades, that is likely to grow in a big way. Now, climate change and plant pathology, research that is being taken up in this way. Yes. Yeah. Yes, climate change. No, this is intersection of the climate change, what we see in plant pathology, that is evolved from traditional prediction to global shift, that is we have seen. It is acting in reshaping the disease triangle, that is now sure. And of course, there are many studies which have been taken up on plant pathogen range, alternating life cycles, and so on. But what we expect, what we need to emphasize, and it will be emphasized, is it is expected to this drastically increase the prevalence, severity, and geographical range of plant diseases that will be studied more and more. It is expected to raise the temperature, of course, in next 2 decades, and elevated temperature affecting life cycle, reproductive rate, that will be studied more and more. And there will be, of course, overwintering and survival of the pathogen that will, these studies will be carrying more weight so far as this is concerned. But particularly aspect, which is important, that is AI integration in plant pathology. This is attracting our attention more and more. That is important, and it is helping to develop tools to combat this unpredictable diseases and disease dynamics that is being changed in shifting climate. AI at present, of course, many mobile apps that has been developed. For example, the plant text that is being used now. And this is AI-based forecasting modules that are already been developed. And potentially this ML, machine learning and deep learning, this will definitely revolutionize our tool, and this is being developed more and more. But now what we expect that AI and machine learning technologies, they are now, of course, new vistas which will be open in plant disease prediction, modeling, and management. Based on symptoms, environmental data, deep learning algorithms, disease prediction models, these are developed and they will be developed more and more. Many models are already there. There are a long list. I have just written few. But CNN, that has taken up in a big way. So this is a neural network-based approaches that has inherent flexibility. It has many features, good features are there. For extraction, for capabilities are there. Significant advancements they have achieved in predictive modeling and predictive plant pathology. And yes. Deep learning, enhanced data rate, what we see in this is that these techniques, they are refined and using in multi-sensor consensus that is being developed in imagery processing. We have seen this deep learning approach that has been applied in many ways in different diseases and for its use. Even mobile apps, these have been now developed to attract this fellow. There is innovative approaches. Those have been resulted for developing a collaboration of cloud-based platform, disease identification, tackling, and forecasting. So these developments are coming in a big way. But what we expect, what is coming more and more, application of artificial intelligence in plant disease prediction, that will be taken up in a major way intervention in the coming years. And in this is expected to shift from reactive detection to proactive, autonomous, and detection in an expanding way. But what will be more in this, that is number one, there will be deep learning and vision transforming. That will be taken in a big way. Multispectral and hyperspectral imaging. It has lots of good potential in identification. But all this, it is expected it will be implemented in a hybrid AI mode. So primary focus that is likely to be on real-time early detection, predictive diagnostics, disease monitoring, multimodal AI, and autonomous systems. These will be taken up. But in this, there are few points that are likely to be taken care of. These are, of course, what we see is that there are certain things which have taken that as an integrated approach. That in many cases, we can see that there will be shift from assisting to autonomous functioning. There will be quick decision making, of course, with this. And it will promote ecological decision this prevention and this expecting breakthrough through this technology. What we see more in this is, of course, there are inherent challenges also. These challenges are certain challenges. They are expected to be minimized. These challenges are like lack of broad spectrum expertise, lack of data sharing coordination, small data sets. These are, of course, needed. And these are a high resource cost that is likely to be reduced. It will draw our attention. And still, there is more awareness that is required to be taken in this way to implement these approaches in plant pathology. So with this, I must say that our colleagues, our students, they are likely to prepare their project. Of course, there are many projects which are now being exported by DST, by even ICR on AI and implementation of AI. AI was a big component in last NA education, this project which was supported by World Bank. Thank you very much for this.
Thank you, Professor Chahal, for excellent presentation. And I wonder how you get time to go through all the literature and collect and present in such a lucious form, you know. It is a very Herculean task you have done. I mean, at this age, you are so very active. So I think you must appreciate, and this is a lesson or eye-opener for the younger scientists, that how he should go for searching for the literature, looking into what others are doing, and then make it in a complete form to present people how it could be done. So it's an excellent idea, and I'm sure we'll be benefited by that. If there is any comment or question from audience, please do so. Because we are running short of time. We have another 2 very important speakers waiting for that. 3. Sorry? Yeah, please.
We are going to start with the award competition, and these are the PR Burma Award for PhD and MSc students. So whosoever is present here, I request them to kindly move on the first floor at the conference hall. Anyone from appearing from PR Burma Award? Okay. And the second award category is of PP Single Memorial Industries and Srimati Guman Devi Memorial Awards. So whoso the contestant, they are requested to kindly move to the basement of this section only. Thank you so much. I'm very sorry, sir, to interrupt
No
you.
mind. Welcome, most welcome. Yes, I think question, no. Okay, our next speaker is a very eminent microbiologist whom I have known for last 4 decades, Dr. Appa Rao Podile. He was the former Vice Chancellor of University of Hyderabad, a very hardcore microbiologist, and still so many students are working under him. He is a mentor for many students who have done so well in their latter career. So Dr. Podile will be speaking on decoding friend and for difference.
Friend and foe. Friend and foe.
Friends and foe. Oh, decoding friends and foe. Differential lipid chain and organic signaling of plant microbe molecular dialogues. You know, he's working all these years on this area for a very long time, and you'll be very highly obliged to listen to him and get some insight. Dr. Podile, please.
Thank you very much, sir. Thank you very much, Professor Mukhopadhyay, Chairman.
And he's also a highly decorated scientist in the field of microbiology.
And I recall, before I start my lecture, I would like to say 2 things. One, it was 1985 in Banaras Hindu University when Professor Mukhopadhyay was chairman of one session in biological control in the Department of Plant Pathology, that conference. I gave a talk as a student. I was in 1985, that was September. And now, after 40 years, again under his chairmanship, in between I have given talks, but he was not chairman during those sessions. After 40 years now, I am giving another talk under his chairmanship. That is a privilege, sir, for me. Thank you so much. And thanks to the organizers for inviting me to share my experiences, research experiences in this particular forum. And the second important point that I wanted to mention is ISMPP is close to our heart for 2 reasons. My teacher, Professor H.C. Dubey, used to have a lot of respect for the society, and naturally, as students of Professor Dubey, we all have a greater affinity to ISMPP, and that is where we have instituted an award in his name, Professor H.C. Dubey, Outstanding Young Scientist Award. And this morning, secretaries have also was talking about it, and I think this has been given for quite some time in the ISMPP. And unfortunately, this 2025 October, my teacher passed away. He is no more, Professor H.C. Dubey. That's just I wanted to
when I am here on an ISMPP platform, I am now recalling that. With this, I would like to move to the topic. The title of it will be a little longer for some of us to read. I just want to make one point in the next 15 minutes. How the patterns matter in recognition of microorganisms by the plant. How the patterns matter. That is something what I wanted to convey to you in this particular lecture. And this pattern recognition helps the plant to recognize who is a friend and who is a foe. And that subtle difference comes here in terms of signaling, where the signal molecule that is coming from microorganism, what kind of a chemical signaling is making that difference to the plant. This is the point which I'm trying to make. We can see here all of them are showing different patterns. Each one of them is showing a different pattern, and we all recognize these patterns quickly. Once we see this, next time when we see this, we don't have any confusion about these patterns, and we can recognize them and happily talk about them. But today, what happened? But today, the patterns are being used extensively. Patterns are being used extensively with this advent of new technologies, whether it is for your mobile recognition, or whether it's your iris recognition, or your face patterns, all of them. Now, if I walk into airport, DigiYatra will not ask me anything if I have registered something, because my photo is already recognized. That's what we are talking. That is the pattern recognition and advanced technology that we are using now for many things. But the plants have been using this pattern recognition for ages. Pattern recognition for ages. Why they were using this pattern recognition? Whether something and a microorganism, because they're living along with several thousands of different types of microorganisms. How to say who is a friend and who is a foe? All of them look same. And that is something that the plants have evolved over a period of time. And excellent talk we heard from Professor Chahal, where he talked about the synthetic immunity systems that he has envisaged in the next 25 years. That is something which can be built on the inherent innate immune system that is there existing in the plant. Inherent innate immune system, where the patterns are actually getting recognized, where there are two types of innate immune, at least two basic systems that we can talk of, where one of it is a very rapid response, which is a broad spectrum response, and other one is a very specific response, which is a little slower, but it is much stronger. These are the two different immune systems that we have. Since we have a limited time, I'm not getting into the details of all of them. I'm ready to discuss with anybody after the talk also. So this is how the recognition happens. What you can see on the right-hand side, the patterns that are coming from microorganisms on the surface of the plant cell, you can see on the right side. The green rectangle is the plant cell. On the top is written as microbe-associated molecular patterns. The different microbe-associated molecular patterns can be recognized by the receptors present on the cell surface. Cell surface receptors, we call them, and they can activate the first layer of immunity, the cell surface immunity, as you can see. But the pathogens also have a strategy to inject some molecules into this plant cell directly, wherein these molecules can, on one side, interfere with the cell surface immunity and thereby induce susceptibility in the plant. That we call it as effector trigger susceptibility. And when the pathogens are producing this kind of effector molecules, the plants also have evolved the second layer of immunity, what we are calling it as resistance-based immunity, or NLRs, intracellular receptors, which recognize these effectors and activate the intracellular immunity. The two intracellular immunity is a stronger one and a robust one, which can work against a specific group of pathogens. Well, I skipped this part. Now, how this is happening in plants, what we see is there are two types of receptor proteins lined on the surface of the plant cell membrane. The first set of proteins have what we call as, on the top panel, you can see they have an apoplastic extracellular domain, and then a transmembrane domain, and below that in the cytosol, you can see an intracellular kinase domain. And the ectodomain recognizes, the extracellular domain recognizes the top one, whatever are written are all the microbial patterns, flagellin or EF2, permease, chitin, extracellular polysaccharide, oligogalactonides, all of them are actually the patterns that are getting recognized. And then the transmembrane domain transmits that signal, and then the intracellular kinase domain activates the MAP kinase pathway, which leads to an immunity in plants. There are second line type of receptors, what we have on the cell membrane, which do not have an intracellular kinase domain. They have an extracellular ectodomain and an intracellular domain, but they don't have an intracellular kinase domain. And what has been known in the recent past is that a receptor which has a kinase domain and a receptor which doesn't have a kinase domain, they come together and form receptor-co-receptor complexes to recognize these patterns. And when these are coming together, there are multiple permutations and combinations are possible for the plant to recognize the varieties of patterns that are coming from microorganisms. And what you see here on the left, you see this kind of pattern recognition is not unique only into the plants. It is also there in animals. It is also there in humans. It's a bit complicated slide. If I had time, I would have explained more. But what I want you to understand is that one of the important signals, one of the important molecules, patterns that are recognized by the plants, because we are talking mostly about fungi, the fungal cell wall material, when it is hydrolyzed into oligomers, chitin oligomers are the ones which are getting recognized. And we all know the chitin oligomers can be generated in the intracellular spaces by the chitinases and gluconases that are produced by the plants and kept in the intracellular spaces. And such type of action results in generation of chito-oligosaccharides, which are recognized by the receptor-co-receptor complexes, what you can see on the right-hand side, and thereby the plant activates its immunity. In parallel, we have microorganisms which are symbiotic, which are friendly with the plant. There is a legume symbiosis, a typical example that we can take. The backbone of the signal or the pattern that is recognized by the plant also has a chitin oligomer. You can see here on the right-hand side, the 4 residues of chitin or N-acetyl glucosamine, which are linked to a lipid molecule, is serving as a nod factor, which induces the nodulation process. One kind of a chitin molecule is inducing a nodulation process. Another type of chitin molecule, chitin oligomer, is inducing immunity. The beauty of it we should see. Now, this is how the nod factor is synthesized, where the chitin synthases are very important in synthesizing, but then they have a decoration of an R group, which is consisting of a nod factor. That means a lipid coming onto a chitin. A lipid coming onto a chitin makes this difference. And mycorrhiza, also another important symbiotic fungus, where you also see mycorrhizal factors, MIC factors. Nod factor and MIC factor chemically are identical. They may have minor variations in terms of the substitutions that they have, but they are backbone-wise and the structure-wise, they are same. The chitin oligomer decorated with a lipid. And if you look at this picture on the top and the bottom, you will realize that the process by which the symbiosis happens, whether it is with rhizobium or whether it is with mycorrhizal fungus, is identical. You don't find much of a difference. The response from the plant is same. The kind of molecules that attract the pathogen microorganisms to the closer to the plant are also same. This is the backbone I was talking about. All of them, if you look at the top of it, it is a generic one, generic lipid chitin oligosaccharide, mycorrhizal lipid chitin oligosaccharides in the middle, and at the bottom you have the rhizobial lipid chitin oligosaccharides. If we just look at these pictures, all of them look same. That means the pattern that is recognized by the plants to enter into symbiosis are similar. Now the difference I'm trying to show to you, on the right-hand side, you see you see at the top panel, LCO is shown, the lipid chitin oligosaccharide. The more of a lipid chitin oligosaccharide, the plant is responding in a symbiotic pathway. And more of the chitin oligosaccharide, as you see on the right-hand side top of the panel, only chitin oligosaccharide without lipid is activating immune pathway. So this is how the nod factor and MIC factor are recognized as symbiotic partners, and chitin alone is recognized as a pathogen, as a factor coming from the pathogen. I just want to highlight in the next 3-4 minutes what we have done around that pattern recognition. One of the things that we have been working is about the chitin degradation by bacterial systems. And at the center, you see an enzyme which we have discovered, where the enzyme takes the smaller oligosaccharides and synthesizes the larger oligosaccharides. And this is the DA, all that what is shown at the bottom is about the complexity of the chitin oligomers that you can synthesize. Since the time is less, I don't want to spend more on that. I can explain. The chitin, for those who work with chitin or chitin oligomers, there is a lot of importance, lot of variations in the chemistry of the chitin. We have to explain chitin in terms of degree of polymerization, the DP that I have shown, 1, 2, or 3, or 4, or 5 residues, and the degree of acetylation, because when chitin is deacetylated, it becomes chitosan. And the degree of deacetylation determines whether it is a chitin or a chitosan. And now you see the degree of acetylation, we also express in terms of fraction of acetylation. Now the third dimension that we have added in the recent last 6-7 years is the pattern of acetylation, where how these acetylated and deacetylated residues are distributed as a pattern. That's what the question that we have asked is whether the difference in the pattern of acetylation is going to make a difference for the plant in recognizing this pattern. And for this, we have used this enzyme, the enzyme which I was talking about, and we synthesized with different types of oligomers. And then next, these are all the multiple chitinases that we have characterized. All these students, as Professor Mukhopadhyay was telling, so many of them have worked. Only these students, all these students whom I have listed, they all have worked only with chitin degradation and chitosan oligomer synthesis and testing of these oligomers. I'm not showing those who have worked with PGPR. All of them, they have created a mutant library of mutant enzymes, and because we wanted to generate different patterned oligosaccharides. And we developed our procedures to prepare these oligosaccharides and tested these oligosaccharides also on by 2 different treatments, like foliar application or a root dip treatment. And we tested about the induction of immunity in terms of these accumulation of phytoalexins, activation of PR proteins, jasmonic acid production, lignification or synthesis of protease inhibitors, all of that. And now, making use of the background that we have generated, taking different chitinases and chitosanases. We prepared a mixture of oligosaccharides, and first we tested it sometime about 10 years ago. We tested them in a cell suspension culture, whether they are able to do the priming in plants or not. And we continued this part, advancing it with multiple enzyme-based methodologies. We try to we make use of different microbial chitin deacetylases. It may look so simple with this slide. It took about the work of about 5 students in about 2 years. 5 students, 2 years to generate these 4 different types of molecules. But to answer the question whether the pattern that is on the chitin oligomer, whether it is going to make a difference for the plant or not, we have generated this kind of molecules. You can see here on the right hand side the histograms, the differently patterned acetylated, deacetylated residues have differential effect in inducing the priming in the plant. That is what that we have published sometime 5 years ago in Journal of American Chemical Society journal. And this is a further continuation. What we showed was a monoacetylated chitin tetramer having glucanac at the non-reducing end has the highest priming activity. That means there is a precise way in which the plants are recognizing the patterns that are emerging from microorganisms. First thing I told you broadly that a chitin oligosaccharide is recognized as a fungus, and by the plant is exhibiting immunity, and whereas a chitin oligosaccharide decorated with lipid is recognized as a symbiotic signal, and then symbiotic pathway is getting activated there. And here we are now showing even more precision is also seen, and the receptor involved in this process is being identified in our laboratory jointly with another collaborator in Germany. These are all the students who have contributed, and these are all the funding agencies which have helped us. And on the extreme right, to my right and to your left, are my collaborators. One of them is, of course, my teacher, Professor T.P. Singh at All India Institute of Medical Science, a distinguished professor for life working. He helped us in structural biology when we were in need of that work. And the other two are chemistry professors, M.J. Swami and Lalita Guruprasad. And Bruno Moschbacher is my collaborator from Germany. And these are all the students who have contributed to this work. And we have carried out this work through 2 European Union projects. One is the 6th framework and the 7th framework European Union project, and DFG funding, and my JC Bose Fellowship and Tata Innovation Fellowship. As I said, almost about a dozen students have worked on in my lab exclusively on this process, and therefore a lot of funding has gone into it, and all these funding agencies I would like to thank, and I would like to thank all of you. I would like to thank ISMPP and Dr. Shalini Singh for inviting me here. Of course, Professor H.B. Singh is responsible for pushing me to come here to Lucknow for this particular conference, and thanks to everyone. Thank you, sir.
Thank you, Professor Pudire, for excellent presentation. And he's a teacher of excellent directive quality and influencing the students in a big way. You know, I've known from people in his universities that most of the students, they want to work under him because they not only get a good training, but also get a good mentorship from all the way to come. So I think the way he presented, the way he showed that so many students are working and learning the experiments and the methodology and the things, and also attracting funds from various sources, speaks a volume about his contribution, you know. And I'm sure you are all impressed, and you should be able to follow as a teacher how to treat the student, how to build them up. Now, any question or comment from the audience you'd like to have? No comment, no question. Dr. Umesha? No, he's not there.
She has something.
Yes. Yes, please. Come forward. You can use the microphone.
Sir, thank you for this insightful session. Sir, as a budding researcher, I just want to ask a very basic question that why did you go for the chitosine and not other lipid molecule? Yeah.
Majority of the pathogens which cause diseases in plants are fungi. And recognition of the fungus by the plant has to happen by recognizing the molecules which are there on the surface when it has to recognize. The first and foremost layer that the plant encounters from the pathogen side is the chitinous cell wall. Therefore, chitin, it is logical to work with chitin, and therefore we started, and also many things are known with that chitin. And that's a very simple reason. There's no other reason. If we are working with a bacterial pathogen, we would have worked with the peptidoglycan. That is also a pattern which is recognized by the plant.
Yep. Thank you.
So thank you, Professor Podere, for your excellent presentation. And I think we'll be waiting for the last speaker, Dr. C.D. Mai. Dr. Mai, please. Hello. Yes. Dr. C.D. Mai is a very eminent personality in the field of plant protection. And not only plant protection, he is known for his work on cotton. He had been Vice Chancellor, he had been Chairman of the Agriculture Scientist Recruitment Board. Now he's doing good work for the cotton. And you know that he has been recently awarded a special award by in Germany, which I read only last night in the Facebook post. So we are very fortunate to have him with us. He is a strong supporter of ISMPP. In fact, they are still going strong and trying to see that the Indian Society of Mycology and Plant Pathology go in a long way. Dr. Mai will speak on how AI, sensor technology are changing dynamics of perfecting and monitoring of pests and diseases. Because these days we are talking a lot about AI, especially in India, everywhere you heard about AI. So what is its role, what is going to play in plant protection? That is what Professor Mai is going to present. Dr. Mai, please.
Thank you very much. एक कॉन्फ्रेंस में मेरा लेक्चर था तो 3 ही आदमी बचे थे, तो आज देव की बात बोले आपके बाद में मेरा नंबर है इसीलिए मैं रुका हूँ, otherwise it becomes very difficult. So I am happy that at least there are a good number of our scientists are still interested to learn. I think you have already heard about AI and sensor technologies from Mr. Dr. Chahal, which was a more theoretical lecture, because what is going on in all the world, it's a good, knowledgeable. Now, what I am telling you is what we have done it in India, my cotton experience. That is what we would like. I am still working with some of the cotton scientists in CICR, and I thought that I will give you that experience of my own, how we developed actual detection and diagnostic and monitoring tools for cotton diseases and pests in India. So this is a very brief, these things what I am going to talk about. I will not spend more than 20 to 22 minutes as allocated to me. So basically, I will just show you, because you're all not from cotton area. So I thought that we will show about what is cotton situation currently, current management practices, what are traditional detection systems, and what are the sensor based. So this is the actually talk about. Probably you all know cotton is one of the most third important crop in India, grown almost 12.5 million hectares. And it is grown in all 9 states of India, Punjab, Rajasthan, Haryana, in central India, Maharashtra, Gujarat, Madhya Pradesh, and in South India, Karnataka, Telangana, Andhra Pradesh, and Tamil Nadu. So this is the situation, and all situations, 60% of the cotton is dry land, and that is where the production is a problem. You can see the output, how it is changing over a period of time, mainly because of the fact that cotton area, cotton pests are creating lots of problems. And you see, once we were exporting nearly 70 lakh bales, now today we have come down to import of 40 lakh bales, a situation which fluctuates in cotton very greatly. And that's the main reason is plant protection in cotton. It is because of the pest and diseases in cotton that is creating problem for us. So these are some of those pests which I thought of: pink bollworm, sucking pest. Then there are of course some boll rot, grey mildew, leaf spot. These are some of those diseases which are. Now our basic emphasis has been the current management practices. What we follow is some of the non-chemical one, like clean cultivation, crop rotation, then monitoring of the incidence and all those things, trichogramma, biocontrol agents and other things. Even, for example, now we introduced a new system called as mating disruption technology, which is very common now. And this was demonstrated by us, myself and our system, SABC, where I'm working, on nearly 7,000 hectares of land in Telangana, Maharashtra, Andhra Pradesh, and entire North India. And now this technique where we have a small wire, where we have a pheromone there, it is tagged on the cotton plant. You can see my colleague is tagging it. I think there is another better slide. Then you can see a small wire, like we call it as Raksha Bandhan of cotton. Very simple. You have that pheromone there where there is a mating disruption. When the male will come for a wrong female and he will find there is no female, so it gets some problems of neurology and then it dies. So this is how the technology we have demonstrated. And now in the central India, pig bollworm problem has been really wonderfully controlled excellently without any much of the chemical use and all that. So these are some of the mass scales there. Now there are of course chemicals, because chemical cotton, you all know that nearly 30% of the total pesticides are sprayed on cotton alone, in spite of the fact that cotton area is only 6% in the country. So this is where our problem is. Now the current detection and monitoring system, we always tell farmers that you use some of those small traps where count the moths in the trap at the least once in a week physically. And when there are 7 to 8 moths per trap per night, it is called as ETL level. Then you go on for spray. This is how we suggest them. Now, in case of bollworm infestation, we say weekly random boll inspection of 50 bolls and then ETL about 10% infestation, spray as per recommendation. So these are the normal recommendations, because we thought that they should not blindly, because most of the farmers are blindly spraying without any kind of, we try to teach them all IPM technologies. We have been teaching them like this. Now the issue is, therefore, this is, you can see how these traps are there. But there are a lot of challenges in these things. One is that the sticky trap monitoring used for estimating pest density, it is sometimes not correctly analyzed. Then manual counting process is always tedious. It is always time consuming, inaccurate, and bottlenecks are there. So as I think shown by Dr. Chahal Saab, there are technologies now which are available, which are very good. So we thought that when all other countries are already using it, why not we try it in cotton? So basically there has been one app developed in cotton, physical and automatic lab, which is installed pheromone traps in the field, and then the solution offered in the AN technology. We have 3 kinds of mechanisms which were developed. First, I will show you the cotton egg app. It's a very simple app where you can, the farmers has to just send a picture of those traps, and this is analyzed and they will say whether you have to spray or not. Very simple. This is already done by a private company also and now available for farmers. Now, we thought that instead of using this, why not we have an entire mechanism? Why not only the app? Why not establish a mechanism? Therefore, what we did it in Central Institute for Cotton Research, we developed a system called as Cotton Microclimate and Insect Monitoring System we have developed. Now here there are 3 components of that. You can see the diagram. The first component is the one which is we have established for microclimate analysis. And then below that microclimate analysis, we have 2 kinds of stickers. One is for the sucking pest complex, which is very serious, and the other is for pig bollworm complex. So there is a continuous monitoring on this system, and then these data are fed to basically clouds. And we are trying to see through Microsoft to get that entire data analyzed and send it to the farmer on their mobile, so that farmers has not to do anything. Only he has to establish that he is using our this micro system, so that he. Now advantage in this is we also analyze the microclimate in this system, and this microclimate you can get the complete. For example, you can see how this structure is there. This is the actual wind sensor is there. There is a light sensor. There is a solar panel. temperature control it. And then on the left side, you will find modified pheromone traps are where they are, you get in the field. Now this has to be in around 5 kilometers area of a village. Once you set up this thing, that 5 to 10 kilometers area in that area, the farmers need not worry about it because they will get information on their mobile. We will monitor everything. We have a dashboard. That dashboard will get all information. And then we have dashboard where the farmers' fields are already noted. And those fields, we immediately say that in his field, there is a more problem. Immediately it will be communicated. So this system which we have, it's a very complex system. We have actually used a lot of this thing like cameras. Then we have these are sensor, high optical lenses are required. This entire thing cost 25,000 rupees initially, or maybe about 40,000. But 40,000 rupees, we are able to do it in 10 kilometers. So if a farmer share this amount, I think he need not worry about any prediction system in his own. So this is how we have developed one system. Now these algorithms employed for image processing identification also we have done ourselves. As he has said, the algorithm is one of the most important part of any system, unless we develop it. And that is all based on correct data analysis. If I know that this is the proper temperature, proper humidity required for any incident, if that data is not fed in algorithm, your algorithms will go waste. That's why we have to have correct algorithms developed based on the data that we actually generate. And this is how the mechanism. Say, for example, we have now cloud servers where receiving the field data from the sensor device, running predictive model. Then we have facilities for web hosting, data backup, data security, and also communication transfer. So this communication transfer, this is the historical data, how it is all converted and displayed and communicated to them. Now we are all thinking in this term that if my institute or any KVK is there, can I do this installation in KVK and then communicate? Because there are intensive areas. For example, I come from Vidarbha. Vidarbha has the largest, nearly 1/3 of the cotton area of the country is in Vidarbha. So that dry land, again, it is all rain-fed. So based on this, we can say, for example, now how image captures are there for ping bollworm and for the sucking pest, and then how this accurately can be communicated to them. This we have practically done it, and now we are implementing it right in the fields in certain villages, which I will show you in the end, which villages we have already done this thing. Accuracy of this also is validated, so there is no problem. Image capture, image data analysis of sucking pest, and what is this availability? We have also done it for ping bollworm. Yeah. And what are the alerts which can be given out of this are also is mentioned here. Now, this applicability of this model is predicting favorable weather and coupling CMIMS with phenology model with the growth of the plant. So this can be area-wide IPM where we use cloud server enables sending information over a very large area that can be sent. This is how this model is there. ICAR has also done one very good job. Actually, the gentleman who has developed is already now, this is actually patented, the system. This is called as smart pheromone traps. And these smart pheromone traps are developed by a Coimbatore scientist, one entomologist. I just wanted to show you how our own people are doing now these jobs. Now this is a schematic diagram, how the trap is there and all that. This trap was developed by them and installed in all farmers' field in Punjab. This is, you can see all farmers' field in Punjab, they have. So this has a 5-kilometer range. Again, this will cost, I think, more than about 30,000 rupees. But then they are all happy that they are able, because ping bollworm has become a serious problem in Punjab, north area. So there it is now predicted, and I am happy that this particular job or this one has been actually, pest monitoring system has 90% accuracy, and also this award is given to one Ramesh scientist, our own. These are the scientists who are associated with Cotton Institute, where they have wireless smart trap for automated pest monitoring cotton was given a special kind of this thing. So this is how our own scientists are able to do it. So my point is that instead of talking more theoretical, now let us implement, and that too you take up the issue which is more serious. As I think somebody was telling in the first lecture, it was telling which is the serious problem in a particular crop. Let us target that crop, then it will become more easy. Now you can see this is what the development of the web portal for access of AI trap data, and the farmer themselves are showing your mobile. He will show immediately, he will come to know whether there is this thing. So there are that my point of telling is only here that how it is possible to really generate. Timely PWD management is becoming very essential. Otherwise, you will have to go on spraying because the pest goes inside the bowl, and once it goes inside, it becomes difficult. So that's why you will have to do it pretty early so that you can use minimum amount of insecticide to manage it. As present, there is no resistance available. Now, a smart portable system is also for diseases. This is developed with my own colleagues in CICR. I am still working with some of my own friends because I was director there. They are at least, they don't say no to me when I go to the institute. Otherwise, most of the directors are not allowed to go to institute once they retire. So at least for me, there has not been a problem. And I go there. So this is a very simple box we have developed. In this box, it is an image. There are lots of what I call it as image albums are kept of all those diseases. At least 3 diseases we are targeting: boll rot and grey mildew and some target spots. So these are once these images are analyzed and see that, so this instrument at present appears to be a little bigger one to carry them in the field. But I think in future we are trying to see that it will become handy like a mobile. And then we install all those images. This is today, if you see, the size is a little bigger one. But this is for very good diseases, and image acquisition is there, image pre-processing is there, feature extraction there, model training can be done, and then the data can be really passed on to farmers. This is how we are trying for disease also. You can see this one, cotton disease identification system, which we have developed. It is called as CODIS in short form. So this CODIS is a very good system which was very recently launched. In the last week, we have launched it at the hands of DDG in our cotton workshop. So this has a lot of utility, and I think in future we are under testing it more with the farmers now. So these are some of those grades. The data are already feed as albums of these diseases. And here also again, we are using microclimate systems to analyze the data. There are lots of other applications like smart irrigation. You can have micro irrigation, drip irrigation and all that. I think many farmers in Maharashtra, there is already advanced. There are sugarcane farmers who are, thousands of farmers are using AI directly in their field. And Baramati has been now in the model. Even our Honorable Prime Minister and Minister of Agriculture, they went to see how thousands of farmers are using it, AI, with no knowledge. They are all illiterate. But how it is all done through a KVK system, which they analyze. They have their own dashboards where the information is passed on their mobile. So there are lots of other applications which are possible under Indian conditions. As I said, I think Chahal has already said that it is the challenges are there. We'll have to pay for Microsoft if we have to go on a very larger scale. And it's not easy because the cost will be very high to put up the data on the clouds and everywhere. So these are some of those issues which are, there is a paradigm shift in India now because of AI. I think everybody is talking about AI, but AI talking will not do. हमने जैसे कहा ना, कॉन्फ्रेंस में हमारा पहला भाषण रहता, बाद में भोजन और बाद में भ्रमण. तो ये तीनों जो छोड़ के we'll have to do something which is concretely done. Then only you will be able to do that. My last slide is basically, these are all some of those problems. There is a village we have adopted very near to Nagpur. It's called as Satnauri. Satnauri means 7 wives. In English, it is called 7 wives. The village name itself is 7 wives. There are a lot of ladies there. We have trained these ladies, and now you can see they are all very happy. The entire cotton system, it was already flagged in Times of India how AI is used by illiterate ladies to manage pest, diseases and water in that village. So I thought that because this year is an international year for women farmers, I would like to devote my talk to them, those ladies. Today the ladies are very less, but I think I would like to devote that talk to them. Thanks very much for giving me this opportunity to be with you. Hope I have not exceeded much time. Dr. Chairman, sir.
Yeah, I can say it is simply superb. Dr. Mai has crossed 80, and you can see his interest, his zeal to work in cotton and going back to the farmer's level, taking all informations. So this speaks a volume about what we should do as a scientist. A scientist never retires, and he is now looking after cotton, go so in depth and tell us how AI can help them. So this is simply eye-opener for all of us, and I congratulate you, and I God give you good health to work further for all the coming years to come. Yeah. So if there is any comment or yeah, patent.
Last one. He's already patented. Yeah. Okay, thank you.
So I think there is no question or comment from the audience further. But I must say that overall, the 4 speakers in the session was excellent, starting from Dr. Garrett from USA, Dr. Chahal, Dr. Apparao, Dr. Mai. They have done justice, but I am only little upset because there should have been more audience. In fact, for such lecture, the hall should be full. I still remember my days in BHU when I was a student, when I was doing my MSc or PhD. When you listen that some speaker is coming, you will see that that time there was no big auditorium or hall, but we'll sit on the floor. We'll sit on the floor to listen and to see the scientist who is working like this. But that's interest or that sort of sensitivity is lost over the time, And I think the organizers should have given a lot of publicity about these speakers, their eminence, so that the more of the PhD students they can come. Because listening to them is a great thing which you'll miss for all your life, So, Dr. Salini, so please remember for future in your conference and other thing, give wide publicity. Tell the students that you should come and listen to him, See, in our time we used to sit. I remember Jant Nardikar, who is no more. He came to BHU to give a lecture. I was at that time in MSc final year. The hall was full. We were sitting in the floor. Still students were standing at the door. That was the type of reaction which you'll have, So I think I'm very proud that my colleagues like Dr. Chahal, Dr. Mai, Dr. Apparao, they're still doing such a wonderful job. And this will be an eye-opener, and not like the current scientists, I'm sorry to give this comment, who really don't go in depth, who really look into the abstract through online, prepare good papers, but they have no time to visit farmers and see their plight. So ladies and gentlemen, I thank you very much for your patience, for your kindness. And I also thank organizer for giving me the opportunity to chair this important session where most of my friends, they have done a wonderful job. So God bless all of you. Thank you very much.
I'm highly thankful to the organizers that I have been given the opportunity to or co-chair the session and listen to the celebrities of plant pathology in our country. If I talk about all of these, Chairman Sir, Dr. Mukhopadhyay, he is father of biological control in India. Dr. Chahal, he is a strategic plant pathologist who has say in UGC and ICR. I think there are few administrators and strategic administrators who has this honor to them. Dr. C.D. Mai, I still remember from my PhD days when he was chairman of the committee for the ICR Senior Fellowship. If I am sitting here today here, it is because of him, because he offered me fellowship, senior fellowship for my PhD studies. And his book, Pathometry by Mai and Datar, it's a classic book in the history. My aim is to tell the younger scientists that we should follow their footprints. We can't be Chahal sir, Mai sir, or Mukhopadhyay sir, but still we should try our best to contribute to the society, particularly our innocent clientele farmers. Thank you very much.
Thank you, sir. Thank you, sir. I now request Dr. Shalini Singh Viseen, ma'am, to please come up on the stage and felicitate our guests. I now request Dr. Shalini Singh, ma'am, to felicitate the chair of the session, Dr. A.N. Mukhopadhyay.
Sir, our Mr. Sahu is a very perfect photographer. You can't move your head now. This is not the good position which you are taking. Sir, with all my best wishes to you that you are here, sir.
May I now request ma'am to please felicitate Dr. A.K. Basant Rai with a token of appreciation and shawl.
Thank you so much, sir.
So it was indeed a very enriching session for all of us. And in the same trail, we now call, ma'am. I now request ma'am to please felicitate our speaker, Dr. S.S. Chahal, sir. आप नीचे आ- Sure. बुला लीजिए ऊपर हाँ जी चालेल So we'll do it for all, sir. We'll felicitate all the speakers. So it's an honor for us.
Sir, please join us. Sir, please join
Yeah,
us.
I request sir also to please join. Shalini ma'am.
All the two, all the.
And Sean. Ma'am, I request you to please felicitate our keynote lecturers, Dr. S.S. Chehel, Karen A. Garrett, Professor Dr. Apparao Podile, Dr. C.D. Mai, sir. Thank you, sir. It is a pleasure that Dr. Karen is also with us online. Ma'am, Dr. Karen is also with us online. So.
Yes, thanks. A pleasure for me.
Yeah. Ma'am, as a token of appreciation, we have shawl and souvenir for you also, Karen ma'am. And we'll try to reach out to you.
Oh, thank you. Thank you very much.
And this shawl is actually a specialty of Benares. So.
Oh, thank you. Yeah, they look beautiful.
Yeah. Yeah, it is about, it's about ODOP. I think our speakers had often talked about it. It is one district, one product. So, yeah, it's a specialty of Benares and Uttar Pradesh as well. Yeah.
Yeah, very beautiful. Thank you.
Thank you. Thank you so much, sir.
Next session.
We'll be now starting with our memorial award lectures.
There are a few guests who have to leave early. Then we will start with this session. Dr. Prashant has to leave.
Okay. Okay.
Memorial award is the baat kar.
Achha, sure. So as few of her, thank you so much.
Great job.
Thank you. Thank you.